Some great questions. So let's talk about those questions because that is definitely one of the first things I want to talk about. The first has to do with the amount of time that will be available for the exams. And again, it is going to be a timed exam. That is a necessity of this. And how much time will be definitely determined by the number of questions. There will definitely be more questions on this exam. The formats of the exams will be slightly different as well. Again, because now we will be using the full proctoring system. It will not only be just recording what's going on your desktop, but also there will be video and audio recording of you and your environment. So that gives us a little bit more flexibility for the time. But primarily the time is going to be determined by the number of questions. Like I said, for the lab exam is going to run the same as the last lab exam did. The only difference will be more questions. So you'll have more time because there's more questions on it. But again, the timing is not going to be per question. It's going to be for the whole exam. So it's really beholden to you to pay attention to that time as you are going through the exam. Again, we'll use simple numbers. If there's 70 questions on the exam, each question will have somewhere between 45 seconds to a minute for a question for you to be able to answer that and type out your answer. So if you're on question 10 and you're 2 minutes in, then you're doing really well on your time and also probably going a little too fast. On the same token, if you're on question 10 and you're already 35 minutes in, then you're going to need to pick up the pace. So it is really going to be on you to pay attention to your time. There is a timer, although I know that timer does freak people out, so you do have the option to turn it off, but I think it is important to keep track of it. For the lab exam, like the last time, just from a logistics and a computer necessity standpoint, because there's probably going to be something close to 70 questions on this exam, I can't have 70 large pictures all trying to populate to your screen at one time. So it will be presented one question at a time. You must answer that question and then submit that question. And once you submit a question, you cannot go back to it. So for the lab exam, and again, just because if you try to go back 20 questions, it can take a long time for those images to populate, and it can be very hard to go through, and it's easier for the computer to lock up, and it's easier for the system to lock up. So just for a safety standpoint, for you to be able to successfully complete the exam in the time allotted, that will be the format for the lab exam. However, the lecture exams will be more like they are in the classroom. Because they're primarily going to be essay questions, although there may be some multiple choice on here as well this time. You will receive the entire test and all the questions at once, so you can look at one and be thinking about one while you answer the other and go back and forth. So for the lecture exam, all of those will be present and all of those will be available at the same time. And again, you'll have a time limit appropriate for the amount of writing that I expect you to be able to do. And that's solely going to be dependent on the number of questions. But yes, I expect both exams to be worth more points than the previous one, so that you should have more time for that as well. The second question that I got before we got started was about the, so again, with the exam, you must have a camera, you must have a microphone, they must be working to be able to do that. I have the practice exam, which looks like I forgot to put on here, so let's put that under the assignments. There is that new, because you guys have already had one practice exam with full proctoring. It is due by Friday, I think at 10 p.m., 6 p.m., something like that. I'm not sure. Due Friday. And if you complete it on time before the due date, then you get 5 points of extra credit. So make sure you do that. Again, it is stupid simple questions. It is a simple 5-question thing. It's the format of the lab exam, so you get one question at a time. And they're randomized, but the point isn't the questions. The point isn't even the 5 points of extra credit. It is an opportunity for you to experience what that full proctoring activity and make sure your system is compatible with it. If your system isn't compatible with it, then unfortunately, there are at least a couple good options, really one good option. That best option is to take the excused withdrawal. The excused withdrawals are being provided. Here's the information that I can definitely guarantee you is correct. The first thing about the expired pardon me, the excused withdrawal is that it does not affect your grade at all. And when you retake the class, it goes away entirely. So it's not like a W where it stays there on your record, it goes away entirely. The second thing I know for a fact, 100% certainty, is that, you get a full refund on your tuition, out-of-pocket tuition. So anything that you paid out of pocket for tuition, like ball waivers and things like that, don't count. But anything that came out of pocket for tuition, you get back. The thing that I am mostly certain about, because everybody tells me it's the case, but I have not actually gotten full confirmation, is that it doesn't count as one of your 3 attempts. It makes sense that that is the case. That is what everybody tells me is going to be the case, but I want to be for full disclosure. I have not been told by an administrator that that is going to be the case. That may be information that is available for you on the ARC website, so that's something that you want to look into when you go through and do the withdrawal. The second piece of information that, again, I have been told, but I do not know with 100% certainty is correct, is that the timing for the excused withdrawal is the end of the semester. As far as I have seen, there is not a quote-unquote date for that. So what that means is if I have not submitted the final grades yet, then you have the opportunity to do the excused withdrawal. But I don't know that. You really need to stop doing that. You don't need, before you make that decision, make sure you check with that to make sure that that is accurate and that is correct. All right, let me see what else I have here, because it looks like you guys have been chatting on the text. Again, curved on the exam, it depends on the grades, the expectation. With more points, then it is more likely to be curved. But just, again, this class is about points. Maximize your points. Don't worry about the curve. Maximize your points. The more points you get, the better you're going to do in the class. It's really that simple. So again, for the lecture exam, won't be a matter of being able to go back to the questions. What should happen is all of the questions will populate at once. So you'll be able to see all, 20 questions or whatever it is that are there. And so you can answer them in any order that you want, and then you just submit once. For the lab exam, it's going to be the exact same format that you guys did last time, 1 question at a time, and you cannot go back. So again, technically you can't go back in either test, but in the lecture exam, all of the questions populate at once. And so you have all the questions there. So you answer all the questions in whatever order you want, and then you submit at once. No, I'm not grading the practice exam for correctness. Anybody who does it gets the 5 points, even if they don't answer the questions correctly, as assuming there is a correct answer. And of course there are for all of them. But no, you complete it, you get your 5 points of extra credit. If, again, a couple of the questions, like the multiple choice questions, there is correct answers and incorrect answers, and so you may get a partial grade for that. 2 of them that are fill in the blank are things that I need to go in and grade manually, the things that you're typing out. So that may be why you're not showing the 5 points, and I just haven't bothered going in and changing all of the grades yet. But even if you get one of the multiple choice wrong, you will still get the point for it. So everybody who completes the practice exam gets 5 points of extra credit for that. I think that is all of the questions. All right, excellent. So that is the game plan. So again, I know there are some technical issues with this for some people, but I will remind you that when we do the exam, and again, up to this point, I have been saying if, but all indications from everything that came out this weekend is the expectation is that we are going to be completing this semester online. So that is going to be the expectation for that. In fact, if you look, they're starting to work on the summer schedule. My understanding is that all of the summer classes that are going to be offered are all going to be online classes as well. So at this point, my expectation is that, again, things can change dynamically. And again, we still have that May 1st date for coming back. Or being let out of quarantine or whatever it is, depending on how things go. So things may change, but the way they always do these things is you plan for the worst and hope for the best. So the planning for the worst is that we will complete this entire semester online, and like I said, summer would be online as well. Let me answer this second question first, the next 2. So Again, there's a tremendous amount of debate as to what is going to be available on the summer, because they're going to a full online summer format. What I will tell you right now is that Cosumnes River, Sac City, and Folsom Lake have all decided that they will not be doing any online lab summer classes. ARC is still in discussion. So there may be some limited micro and A&P that is available online this summer. But as you've seen how challenging this can be, taking this online when you only have to come to class twice a week and then you have 5 days to study it, don't take these classes online. I teach the online class, I teach the summer class on campus, and I spend the first hour of that class, and this is not an exaggeration, I spend the first hour of that class telling everybody who's there to take A&P during the summer that they are stupid for taking A&P during the summer. The average grade during the summer is worse, almost a full half grade worse than it is during the normal semester. And as I've been trying to beat into your heads, this information in 430 isn't just randomly selected. I am picking things and information that you need not just for the rest of 430, but also for 431. And the retention of that information going from the fast pace of summer into the fall is horrible. So typically the students, not only do they do worse during the summer class that they're taking, but when they take second semester, they do far worse as well. So I would strongly, strongly encourage you not to take these classes online if they're available. And at this point, I don't know if they're going to be available. Let's go back to the previous question. If the previous exam was our lowest grade, how will you curve that? Again, the logistics of that I haven't figured out, but my guess is that what I would do is go percentage-wise. If percentage-wise your skeletal system exam was your worst exam percentage-wise, then I would replace it with the final percentage-wise as well. So if you got a 70 on your final exam and you had a, or I guess an 80 on the final exam, and you had a 60% or something like that on your skeletal system, then I would replace it that way. But I'm not going to obviously replace a score of 44, which would be really good, on that with a 72 from that. So we would split the difference or something like that. But again, I'm trying to get through day by day at this point, and so I haven't really thought about the logistics of that. But my guess is that's likely what would happen. All righty. Any other questions on the logistics of the exam and what we have going forward? Was it the ARC requirement for the proctoring exams or yours? ARC. So, by ARC, I mean the biology department. In the biology department, everybody in the biology, actually really science. In the science department, these were the requirements that were set up for the appropriate proctoring of these exams. Again, I know other universities have had different discussions and different things, but as a department, it was decided that this was going to be the format that we would accept to maintain the integrity of the class, to maintain the quality of the class. And so, especially once the excused withdrawal was offered, then people had an opportunity where if they either are unable or unwilling to take the exams using this format, that there was a reasonable alternative for them to be able to be able to not be penalized for not being prepared or even not wanting. That's the great thing about it. Ultimately, it was set up to help those people who didn't have the technological requirements for it. But at the same time, as I've said many times, I didn't sign up to teach an online class. You guys didn't sign up to take an online class. And if for whatever reason you're not comfortable being monitored in this way on the exam, then you are not required to do it. So again, so you do have that option of taking the excused withdrawal. Yes, typically the final is all multiple choice questions. Again, I don't know if that would be the case anymore. I may have to change that because of the format. So I haven't thought that far. Again, I haven't thought that far ahead because the week we come back from spring break, I have your lab and lecture exam on Thursday, and I have my 431's lab and lecture exam on Wednesday. So I've just been thinking one exam ahead. in the past, the final exam has all been multiple choice, but my guess is that this time there will be more essay questions or something about that, just because of the different format. But again, I haven't even thought about the finals. I haven't thought about your nervous system. I haven't thought about anything but our muscle exam and the urinary and respiratory exams that are coming up. So I haven't looked that far ahead. All right. So any other questions on what to expect from the exams going forward? These are all great questions. And again, that's why I wanted to take this time at the beginning of the class to make sure that we addressed all these things. Again, that's why I wrote that big, whole, huge treatise that is now the homepage for our Canvas site, just so that everybody fully understands what is expected of them moving forward from this, what the expectations of the class are going to be, so that everybody fully understands and there's no confusion and everybody knows exactly what they need to do to be successful. Because again, that's always my goal. My goal is to help you to be successful. So I am happy to provide you with as much information as I can so that you understand this process, so that ultimately you can be successful. All right, excellent. That we'll get to that next. So, all right. So, with the exam out of the way, then let's talk about the things that are going on today. We are going to continue our discussion of muscle physiology. We finally, in the last class, finished our muscle contraction, and then that process. And so now we can start building back up. Now that we've broken it down and seen how the bits and pieces work, we can slowly start building it back up to some higher level concepts. To help us to understand this, and then of lab, we will continue our discussion of the origins, insertions, and actions. Again, I don't have the opportunity to go through all of them, but I will try to highlight some of the important ones or the confusing ones that I think people are challenged with. So we will talk about that. There are the assignments that you have, the ones that you already know about. The Unit 11 review is due on Thursday, 2 days from today. Your Physio X Exercise 2, which have 7 activities, that is due as well. On April 14th, there will be the same class with information that we should learn. Yes, absolutely. So again, there will be a new information that will be provided on Tuesday, the 14th, 2 days before the exam, which again is not a format that is unfamiliar to us. However, as I should also remind you, this isn't new information that I'm pulling out of the ether. You have your outlines that are on Canvas that show you the things that we're going to talk about. So should you spend this weekend not just studying the stuff that we covered this week, but also looking ahead at next week's lecture? Absolutely, because again, you'll see on there, our primary focus, we're going to get through most of the skeletal system this week. And so next week, we'll finally get to come back and see how, after we've learned everything you ever wanted to know about how a skeletal muscle cell contracts, we will come back and talk briefly, and it will be briefly. Obviously, it's not going to be emphasized as much as the skeletal muscle tissue physiology will, but we do have to talk about smooth muscle and cardiac muscle and how they function and how that function is similar and also different from skeletal muscle. All right, so those are, and so you have the Physio X that's due. You have the Unit 11 that's due. We already talked about the practice exam with the full proctoring that is due on Friday. The other thing that is due on Friday is a new muscle activity handout. As I mentioned, and let's go ahead and switch our view here. So I want to go, where do I want to go? Here. In your module on your Canvas site in section 4, under the lab handouts, there is a new homework assignment, a muscle activities homework. The same way that in our skeletal system, I divided you guys up into groups and we presented the bones and bone features to make the anatomy a more interactive process. I do the same thing with the muscle system. What I do is I give you a list of muscles, and your job is to come up with a common, and commons in quotes there, activity that helps everybody to remember. Basically a mnemonic, a memory tool. And if you fondly remember the lecture as I do, then you should remember the example I gave you when we talked about the sternocleidomastoid, right, where we flex, wait, where's my annotation? There it is. where we talk about when you use both, it flexes the head, or it can elevate the rib cage, or if you just use one, it rotates the head. I told you the story about how I had the student who gave me the example of a bong rip, right, where she lowered the head, inhaled, and then rotated her head to the side to breathe out. So I gave you that as an example because that's usually one of the kind of things that people came up with. And so what I've done here in a handout, this is kind of, again, I'm trying to find ways to encourage you to do the lab portion of this. The lecture part of this class is the easy part. The lab is the challenging part because up to this point, I've been mostly, feeding you guys like baby birds. Now, much more of the onus is on you to study and master this anatomy. And so I'm trying to find tools and ways to do this, and I thought this would be a fun way to do that. So the top part of this, what I've given you here, is a list of activities, common activities that you would do. And again, I made sure that you understood the description of the motion. So that's what I mean. Like, so for instance, if you're doing that freestyle swim stroke, right, where you're bringing the up and moving the arm forward and rotating it around, is that swimming stroke. I've given you these common activities, and you have to figure out what muscle would allow you to do that activity. And then once you have the muscle, of course, write out the actual actions that it has. And then for the second half of this, you are going to come up with the activity for this. So for instance, I've given you a muscle like the scalenes, where it flexes the head and it elevates the ribs. Oh, and I'm missing part of that there. And it laterally flexes the head. So I'm missing that part of that there. So let's go to another one. Pectoralis Minor, where it's going to protract and depress the scapula and also elevate the rib cage. So these are the three actions of the Pectoralis Minor. So you're going to come up with an activity that you would do where you would use that particular muscle to do those things, to do some particular activity. So whatever that is that thinks that you would get up and demonstrate for us, that would be what it is. So like the rhomboids, for simple, it retracts the scapula. So the example I used was you pull your hand back to throw a punch, right? Not the actual throwing of the punch, but the pulling the arm back to do that. All right. So this is your activity. It's a way to force you to think about the muscles in a lateral way to try to make sure that you're understanding and making sense of it. And it's due by Friday. All right. Now, again, this part here, your activity that you're coming up with is up to you. There's really no wrong answer as long as it incorporates all of the actions. For instance, for the internal oblique, you wouldn't want to just say, tightening up your stomach muscles if someone's going to punch you in the stomach. Because while that is what you do when you compress the abdomen, it doesn't do any of the other three actions. So as long as your activity incorporates all the actions of the muscle, then it's a right answer. And there should be only one right answer to all of these as well. So when you're doing these, and again, here's one that should be easy because we talked about it in the last class, figure out what muscle would allow you to do this activity and then write the actual actions of that muscle. And so this is something to really force you to think about the anatomy portion of this, and it's due on Friday. And so again, you can print this out. If you don't want to print this out, you can also just type out the answers on a separate piece of paper. And then if we go back to our modules, like all of our other homework assignments, there is now a location where you can drop that in the Dropbox here. So you'll just click on that assignment and you are going to submit it. It's due by noon on Friday, and you're going to submit the assignment there. Yes, this is something that will be graded for correct answers. So I do want correct answers. This is not just fill in the blank with just whatever random things you think of. I want you to actually think about this. This is a 20-point assignment. Typically, assignments worth more points means that you need to come up with the correct answers because I want you critically thinking about this information. Yes, all of the activities that I've come up with also, great questions. So if we go back to the list, all of my common activities, throwing a bowling ball, all right, raising your leg during a true love kiss, all of these things are off of muscles that are on your list. So again, the whole point of this is to study the things that are on the exam. Yes, so if you want to print it out, take a picture of it and submit it that way, that's fine. If you want to write it out on a piece of paper, that's fine. If you want to scan it as a PDF, that's fine. I purposely have, for all of the homework assignments, and again, not just this one, but for all of the homework assignments, except for the PhysioX. The PhysioXs make PDFs, so it's going to be PDF. But all of the other unit reviews and all of these, I have not limited your file type that you're submitting so that you have as many ways. If it's easiest to take a picture, if it's easiest to scan it, if it's easiest to just type it on your computer and save it as a PDF, you have whatever option is necessary for you to turn those things in. Alrighty, so that is the one new assignment that I've set up. Oh, there we go, perfect. The one new assignment that I've set up. Well, two, because we have that practice exam, but we've already talked about this. This is the second new assignment. So basically 4 assignments. 3 of them are due this week before spring break, and then you have your PhysioX to help you during spring break. Which reminds me of the other thing that I wanted to mention. Next week is spring break. Because it is spring break, there is no lecture. So no lecture next week. However, I appreciate and understand the challenges that are going on in learning this material. So I'm going to have formal office hours on Tuesday and Thursday. From 12 to 2. So during our normal class time, I'm going to be here on the computer, because quite frankly, I'm going to be writing exams anyway. So I will have my computer on. These office hours will not be recorded, so I'm not going to be recording them. But if you have any questions or any concerns or any problems that you're having, or you just need better help understanding the muscles of the forearm, or any of the things that are challenging you, I will be here actively during those times. If those times don't work for you, then also you are, of course, always welcome to email me, and we can converse via email. Or the other thing that we can easily do is set up a time to meet in Zoom. Again, I do have my own Zoom classroom or office, and it's my phone number for my office, 916-484-8992, where we can meet at any time. So I can do that as well. But I will be on the computer. Zoom will be up. I will be able to answer any questions during that time formally as well. All righty. So there you go. Those are all the notifications. That is all the information. That is everything we have. Any questions on any of that? Excellent. The blank stares of the blinking computer tell me that we are good with that. And now we can finally, a half an hour into the class, finally get back into our lecture. Excellent. But again, no, I'm absolutely not complaining. These were all important things and important questions. Like I said, my goal is for you to be successful. This is a brand new format for all of us. Again, I many years ago taught the 100, which is the introductory A&P class, online, but that's a lecture only. And that was way back when we were using Blackboard. So again, that was a billion years ago. So this is, all of this is new to me. I'm sure much of this is new for you guys. Certainly trying to do a lab class online is new. So again, the goal is for everybody to understand what's going on so that everybody has the opportunity to be successful. All right. What did my next slide look like? All right, I think we can sneak in what I want to do here. So when we left off last time, like I said, we had gone through and finally completed all of our 3 big steps of how to get a muscle to contract. We had then went step by step by step by step through the entire process of how the muscle would relax. We put the entire process together into the long, continuous process that would have been our muscle relaxation. We could do the same thing here for the process of the muscle contraction, and that would be useful, but actually it's something I would encourage you to do for yourself as a practice for this. Actually put all 3 steps together, step by step by step, and make sure that you understand that. And that would take a tremendous amount of time, which is why I'm not going to do it. But what I do want to do in this case to remind us about this is do 3 things. What I want to do is I want to, of course, as always, identify the 3 steps. Every time we identify the steps, we always want to think of what the goal is for those steps. But I think the other thing that will help us to understand how these relate to each other is to identify both the first step and the first part and the last part of each individual step. So, of course, if we start with part one, as we've talked about, is communication at the neuromuscular junction. And I am going to abbreviate here just so that we fit in the space. And of course, our goal in this, as we talked about, is to convert a neural action potential into a muscle action potential. Again, I think we've written that enough times where it's okay if I abbreviate that here. But what I would like to do to that now is add, how does this start? So someone remind me, communication at the neuromuscular junction, the actual muscle contraction itself, where does this process ultimately begin?
In your brain?
Absolutely, right. You are absolutely correct. And there's the magical word I was looking for. You make the decision. Or A. You can go with A. I like A. Right? Communication at the neuromuscular junction starts with the process of us making a decision. When we make a decision, we produce that neural action potential. It spreads down, right? That's what we see here with the pretty picture. It's going to spread down to the synaptic end bulb. It is a big, huge positive signal, so that depolarizes the synaptic end bulb, opens up the voltage-gated calcium channels. Calcium rushes in. That calcium rushes in, triggers the exocytosis of our acetylcholine, right? And on and on and on and on. So all the things that go along with that. That is where that process starts. And of course, it ends. Communication at the neuromuscular junction ends with what?
Contraction?
No, that is where, again, that is the goal of this whole entire process. But remember, and you're right, it is a continuous process. Ultimately, it's going to lead to contraction because it is a big continuous process. But remember, we took this process and chopped it up into 3 more easily digestible pieces. So the first piece is communication at the neuromuscular junction. Alina is correct, although I would take it one step forward. It ends when at our motor end plate, we reach threshold and produce a muscle action potential. Right? So absolutely, we reach that threshold, and when we reach threshold, and again, we want to say where that is, at our motor end plate, we reach threshold and we produce a muscle action potential. All right? So that is our communication at the neuromuscular junction. We've talked about our goal. We know our starting point. We know our ending point. That, of course, brings us to step 2. Step 2 is excitation, contraction, coupling. Give myself a little bit more room, sneak it in there. That doesn't quite fit, but that works. Again, we need our goal. And remember, the goal of the excitation contraction coupling, as we see over here on the illustration, is to use calcium to move the regulatory proteins. Where does this process start? Now, it doesn't start with the releasing of the calcium. That's our goal. Our goal is to release the calcium from the sarcoplasmic reticulum. That is absolutely our goal. But where does the process actually start? If I asked you the essay question to describe the process of initiating communication, if I asked you the essay question, how do you initiate excitation, contraction, coupling? True, we need our, there we go. We need our triad to depolarize, but of course what depolarizes it? The muscle action potential. Remember, one of the things we talked about is that this is a continuous process. What that means is that the last step in part 1 is also the first step in part 2. So it starts where the other one ended. So at the motor end plate, we reach threshold and produce, oops, and I'll abbreviate it here for space, muscle action potential. If we wanted to add anything to that, we would then say that is the starting point, but I guess in parentheses, obviously, then that muscle action potential, as you guys talk about, spreads down the sarcolemma. Because again, when we produce that big positive signal, that big huge wave, it doesn't just stay where we put it. But you guys are absolutely right. It travels down the transverse tubules. It travels to the triads. It depolarizes the triads. That opens up the voltage-gated calcium channels, allowing the calcium to come out, where it binds to the troponin, changes the shape of the troponin, rotates the tropomyosin, right? And that is ultimately our endpoint. Our endpoint is when our troponin rotates the tropomyosin off, let's say it that way, let's be specific, off the active binding site on the actin. Right? Because that's our goal. Our goal was to move those regulatory proteins. Of course, that leads to step 3, our contractile cycle. Our contractile cycle obviously has a goal, and that goal, as we've talked about, is to change the shape of the muscle cell, right, or to generate our force, or produce that tension, whatever we want to put there for that. And hopefully this should be obvious now. Step 3 starts where step 2 ends. So our contractile cycle begins with troponin, rotates the tropomyosin off the active binding site on the actin. And does it have an end? Does the contractile cycle have an end? No, exactly. Right. The key word there is cycle. That cycle is the continuous process. So as long as that troponin is moving the tropomyosin out of the way, our contractile cycle will continue. Again, it will not be an essay question. But should you be able to describe this entire continuous process from the point where you make a decision to where the contractile cycle is continuously going around? Absolutely. You should absolutely be able to do that, right? But remember, I'm not going to ask the question that way. I'm going to ask for these individual steps. And in fact, in the last class, if you remember, we came up with the 7 possible essay questions, 2 of which I guarantee will be on the exam. All righty. Questions on this? Again, this part has to make sense. If this part doesn't make sense, then there's not much point in going further. So any questions on any of this before we move forward? All right, excellent. Let's move forward. Now, as I mentioned, once that contractile cycle begins, it's going to be a continuous process. But ultimately, it is going to stop at one point. Otherwise, when you contracted a muscle, that muscle would contract and it would stay contracted forever. And clearly that's not what happens. So instead, what happens is we have a muscle contracts for a period of time until it stops. And the types of things that would cause it to stop is going to be basically three main things. Obviously, as we've already talked about, what started the muscle contraction was the decision. So what is going to finish the muscle contraction is going to be a similar decision. And remember, when we decided to contract, we started producing neural action potentials. So one of the primary factors, okay, we'll go ahead and put them there. One of the primary factors that determines how long a muscle contracts for, really, if you think about it, is the number of neural action potentials. Because the more neural action potentials we produce, the more acetylcholine is going to be released. Right? Acetylcholine is what is necessary to open those cation channels, allow the sodium to come in, and that's what depolarizes our muscle cell. So if you think about it, the more acetylcholine, the more the more muscle action potentials. So if we want to get the muscle to stay contracted, we have to produce more muscle action potentials. But the muscle action potentials by itself is not enough, because there are two things that are necessary for that contractile cycle to continue. The first is we have to have that free calcium right in the sarcoplasm. It has to be released from the sarcoplasmic reticulum, because as we've talked about, our calcium is going to move the regulatory proteins. If we can't move the regulatory proteins, then our contractile cycle can't continue. However, as we also talked about, our contractile cycle can't continue if we don't have ATP. That ATP is the energy for the power stroke, right? The ATP is what causes the myosin to let go of the actin. So if we don't have ATP, we can't continue the cycle as well. So if you think about it, how long a muscle contracts for is really determined by these three main factors. How long are we going to continue to stimulate by our nervous system? Because, and again, this is a concept we'll get to a lot in this class, over the next, this week, I guess. But one of the important things to remember that essentially one neural action potential produces one muscle action potential. And so the more neural action potentials, one neural action potential produces one muscle action potential. So the more neural action potentials we produce, the more muscle action potentials we can produce. And the longer the muscle is going to contract. But to be contracted, we need calcium to move those regulatory proteins. The good news is calcium doesn't get used up. Calcium binds to the troponin and let go, and binds to the troponin and let go. So it isn't that we're using the calcium, but calcium has to be available. Because remember, the cell doesn't want the calcium in it. It has pumps to kick the calcium out of the cell. It has pumps to lock that calcium back into the sarcoplasmic reticulum. So if the cell can get rid of its calcium or get its calcium locked back up, that will stop the muscle contraction. But we're not actually using it up the calcium. ATP we use up. Remember that myosin head breaks the ATP. It doesn't really break it, it splits it. And that's what releases its energy. But when you split an ATP, you don't have an ATP anymore. Instead, you have that ADP in the phosphate. So this is a resource that is used up in this process. And as we talked about, if you run out of this resource, that muscle contraction stops. All right. Now, as we mentioned, when these myosin heads are grabbing and pulling on the actin, they pull the actin towards the M line. So on the right side, it's pulling it towards the M line. On the left side, it's pulling it towards the M line. So our sarcomere gets shorter because that actin is connected to the Z disc. So the right Z disc moves towards the M line, the left Z disc moves towards the M line, and as they move towards the middle, our sarcomere gets shorter. Now, of course, our myofibril is made up of many sarcomeres, and when we release that calcium into the cytosol, it connects to all the regulatory proteins, so all the sarcomeres are going to shorten at the same time. So when they shorten, our whole myofibril gets smaller. Notice also, and here we'll clear our drawing so we can take a look at this, this shortening of the sarcomere causes a change in its anatomy, right? When the muscle contracts, here's going to be the key word, some of the regions of the myofibril are going to change. Let's think about them. If we think of all of the regions of the myofibril, we have the A band, we have the I band, we have the M line, we have the Z discs, or Z line, and we have the H zone. All right, those are the 5 regions that we have there. And some of these are going to change as the muscle contracts. So let's spread these out a little bit more so we have more room to play with. There we go. Move that over here. And then that will allow me to come over here and grab my pointer to talk about this. Now, notice when our sarcomere contracts. Here it is in its a relaxed state. Here it is in the contracted state down here. Remember, our A band is defined by the region that contains the myosin. Has the length of our myosin fiber, this thick filament, has it changed when the muscle contracted? Is the myosin filament the same length in a rested and a relaxed and a contracted sarcomere? No, it doesn't change, absolutely. So again, there is no change. Let's change colors here, though. There is no change in the length of our A band when our muscle contracts, because it is defined by the myosin filament, and the myosin filament doesn't change. But notice what does change. As the actin is being pulled towards the M line, there is more overlap of the myosin and the actin. So actin and myosin are now overlapping more. And that does change 2 regions. Notice the first region it changes right here is the I band. So notice when the muscle cell contracts, the I band is going to get shorter. Before, and again, we didn't, in fairness, didn't see the entire I band over here, but I will tell you, as you've looked at our striped muscles, in a normal resting muscle cell, the I band, the light stripe, and the A band, the dark stripe, are similar in size. Now you can see how the I band has gotten much, much shorter. The I band is the region from one myosin to the other. It's this little bit of space in between, and so our I band gets shorter. Now, notice everything is being pulled towards the M line. So if you think about it, our M line does not move. But what about the Z discs and the Z line? Do they move as a result of this? Yeah. What happens is our Z lines move closer to each other. right? Because essentially, as we talked about, our sarcomere is getting shorter. And they are the walls of the sarcomere. So not surprisingly, they get shorter in relation to each other. Oh, so well. And that leaves us with our H zone. Notice here also our H zone is going to get shorter. And that's what our illustration shows here. Our H zone, and remember the H zone is the amount of, is the location where there is no overlap in the myosin and actin, just myosin alone. And so our H zone gets shorter. However, there's something else really cool that happens here as well. Notice here, and let's go ahead and do this. We see our H band has gotten much, much smaller as a result of this. But as we continue to contract this muscle, the myosin and actin will actually come together. And in fact, and let's go ahead and cheat and draw it. What can actually happen is that the actin from one side can actually connect to and pass through the myomesin. The actin from the other side can actually come to the myomesin and pass through it as well. Notice if that occurs, exactly. If the actin actually gets pulled close enough to the middle, the H band can potentially go away. So our H band can potentially, if we contract the muscle enough, entirely. It can go away entirely if we contract the muscle enough. So absolutely, that's what can happen. What can actually happen here is if we contract the sarcomere enough, the actin from the two sides will touch and interconnect with each other, interact with each other. And when that occurs, the H band goes away entirely. Now, it's easy to see this on an illustration and have that make sense, but that can't be what it really looks like, can it? Yeah, it actually really does. What I love about your textbook, as I've mentioned many times, is it has these great electron microscopy views. Again, this is where they use a super powerful electron microscope. We take a little bit of tissue, you freeze it, you slice it, you put it on a slide, you put it in this machine, and it is bombarded with electrons. During that process, the tissue is destroyed, but what it does allow us to see is these incredibly microscopic views where we can actually see the individual proteins inside of a muscle cell. This is something that we cannot see with any magnification of light microscope. So it is tremendously, these electron microscopes have tremendously increased our understanding of our body and our world and how things work. So here we have the, again, nice illustration of a muscle cell at rest. Our sarcomere, again, our I band and our A band are similar inside. We have a large H zone, an M line at the middle, right? I band with Z discs on the side. And if we actually look at the myofibril inside of a muscle cell, that's what we see. We see dense proteins here forming the Z disc. We see really dense proteins here. This is where the myosin and the actin overlap, right? Notice here is only actin, and it's much, much lighter because remember, actin is a thin filament. Here, we have the part that is only myosin. It is much darker because it is the thick filament. So here we see the thick filament, and of course, this region of the thick filament would be the H zone. Here we see the actin. This is part of the I band. And here we see where the actin and the myosin overlap, much, much darker. And then here at the midline, we see that myomesin that interlocks the myosin, forming our M line. Again, no new information here where you would see this. To answer your question, think of it this way. When you flex your elbow, as you flex your elbow, right, to make a big muscle to show off your guns for, whoever, as you do that, there's a limit to how far you can contract that, right? You can't actually get your wrist to touch your shoulder when you contract your elbow. part of that is obviously the joint of the elbow limits your range of motion. But the other reason why your muscle gets weaker when you are flexing and fully flexing it like that is because you are getting the myosin and actin to interact. So it doesn't actually take any kind of weird or unusual type of contraction. As a muscle contracts, the more the muscle contracts, the more it moves, the more it's going to interact. And actually, that's one of the reasons why our contractions get weaker the more we contract that muscle. We'll actually talk about the tension it can produce. And yeah, when these are overlapping, like we see here, we have a fully contracted sarcomere. Notice in this fully contracted sarcomere, as we mentioned, the actin from both sides is now interwoven in that myomesin. Notice now our H zone has gone away entirely because our myosin and actin, right, or our myosin, I should say, is completely overlapping all of our actin. So notice there's not that, and let's go back, there's that light region of the H zone here, and here it's gone. There is no H zone because now the H band has gone away because our myosin and our actin are completely overlapping. Our actin has intermingled with our myomesin. Notice our I band is much, much smaller. We couldn't see the full range of the I band before, but now we see it and it is much, much shorter. Notice also our Z discs, if we were to measure the distance of this apart here, and let's cheat and do that. We can do that with a line. We can draw a line to show the length of that sarcomere when it is at rest. And then when we switch the page, we can see that our Z discs have moved towards the center and our sarcomere is much, much shorter than it was before. But notice also our M line is still in the middle. It is still unchanged. So again, to emphasize this point, if you think about it, and again, let's do this as well. Change colors. So if you look at our A band, there you go. Notice our A band hasn't changed. Our M line hasn't changed. Those two are not changed by a muscle contraction. But the overall length of the sarcomere, the size of our I bands, the size of our H zone, and the distance that the Z discs are from each other, all of those things change when our muscle cell contracts, when our sarcomere contracts. All righty, questions on that? All right, now, so far we have just been talking about what happens in one sarcomere, but as I already hinted at, all the sarcomeres in a myofibril are going to contract at the same time. Remember, that same muscle action potential spreads down the entire sarcolemma. The sarcolemma is specialized to propagate an action potential, and it propagates down all the transverse tubules. And as it propagates down all the transverse tubules, it is going to stimulate all of the triads, opening up all the sarcoplasmic reticulum, releasing all that calcium. So all the sarcomeres in a myofibril all contract at the same time. So if you notice, as all of the sarcomeres get shorter, then the effect of that is our entire myofibril gets shorter. And remember, this myofibril is connected by structural proteins to the plasma membrane of our muscle cell. So, and they run the entire length of the muscle cell. So when our myofibril gets shorter, then our muscle cell gets shorter. And when our muscle cell gets shorter, we can build up from there, as we talked about. That muscle cell has connective tissues that wrap around it, connecting it to its fascicle. So when the muscle cell gets shorter, the fascicle gets shorter. The fascicle is surrounded by connective tissues that hold it to other fascicles and form the muscle. So when our fascicle gets shorter, the muscle gets shorter. So in this fashion, we can build all the way up from these single proteins, that myosin head grabbing and pulling, leading to the change of the shape of our muscle, moving our body through space. All right. Now that we understand how to change the shape of the muscle, what we need to talk about more is about this concept of the force that it is producing, or more specifically, the tension that it is producing. Because when our muscle cell, and again, remember the term muscle cell and muscle fiber are interchangeable. When our muscle cell or our muscle fiber pulls on that connective tissue, it produces a tension. And that tension is what moves the body through space. However, flexing your forearm, right, flexing your elbow, you can do that to bring your phone up to your forehead. You can do that to bring your glass of milk up to your mouth. You could also do that to lift your bed to get your sweaters out from underneath or something like that. And lifting a glass of milk and lifting your bed both use the same action where you are flexing your elbow, but how much tension you need to do that is going to be very, very different. So while we can flex our elbow many ways, we also need to be able to control how much tension we have in that muscle. Now, as we mentioned, the tension comes from myosin heads pulling on actin. So the more myosin heads pulling on the more actin gives us more tension, right? And so how many myosin heads we have is determined by how big our muscle cells are and how many muscle cells we have. So if you want to lift up your bed, you need to use a lot of muscle cells all firing together to flex that elbow to lift it up, right? And hopefully most of you, all of you, should be able to lift a bed, right? How many of you can lift a car? Probably not super many of you, right? So again, there is a limit to how much tension you can produce, and that limit primarily is how many muscle cells you have attached in that muscle. But how much tension a single muscle cell produces can also vary. Muscle cells obviously have a maximal amount of tension they can produce, but they're not all producing their maximal tension all the time. So what we want to be able to do is modify how much tension a single muscle cell can produce. So in a single muscle cell, we can produce more or we can produce less force. We can produce more or we can produce less tension. And so since we're talking about a single muscle cell now, we've gone from a sarcomere, we've gone from myosin and actin to the myofibril to now a muscle cell. In that muscle cell, there are two primary factors that modify and influence how much tension that muscle can produce. The first is the length of the muscle cell when we contract it. Right, we've kind of already gotten a hint at that when we were seeing how the sarcomere changes when it contracts, and we'll get to that in more detail in a minute. But the second is the frequency of stimulation, right? How many, and again, remember, this is all going to be determined by how many neural action potentials we produce. Because remember, one neural action potential is going to give us one muscle action potential. And so that factor of how much we stimulate it determines how many muscle action potentials we produce and how much tension we're ultimately able to produce as a result of that. All right, so that's our next step. We need to talk about how we modify the tension in a single muscle cell. Now, I know we haven't gotten quite as far in the lecture, but we did a lot of review and a lot of other discussion. So this is a good natural stopping point. So let's go ahead and take our first break here. Real quickly, any questions before I stop the recording and we take our break? All right, which question, Eric? Oh, great question. So the voltage-gated calcium channels located on the sarcoplasm are, and again, not on the sarcoplasm. There's no voltage-gated calcium channels on the sarcoplasm. They're on the sarcoplasmic reticulum, right? So again, the sarcoplasmic reticulum, well, I guess as opposed to sarcolemma, most of them are located near the triad. So if we, let's cheat. Let us in the lecture Here, hold on. Cheat and go. Come on. There we go. If we go way, way back here. Oh, I like that picture. Let's go with that one. Nope, I lost it. Here we go. This one works. Notice here, there's our transverse tubule. Here's our sarcoplasmic reticulum. And again, the sarcoplasmic reticulum and the terminal cisternae. Notice in this case, they emphasize the voltage-gated calcium channels on the terminal cisternae and their proximity to the transverse tubule. But I will tell you that there are actually voltage-gated calcium channels along the entire length of the sarcoplasmic reticulum. There may be more of them here where the terminal cisternae is, because here there's more sarcoplasmic reticulum. And if there's more sarcoplasmic reticulum, there's going to be more calcium. But there will be voltage-gated calcium channels as well as calcium pumps along the entire length of the sarcoplasm. So the other nice place, and actually I was going to do this during the break, but let's go ahead and do it now. Because I'm hoping it's there, but I'll be honest, I haven't actually cheated and looked yet. If we go to that, where is it? Virtual Anatomy Lab. On the Virtual Anatomy Lab under our muscle system, and I'm hoping it would be, oh, they're not going to have it, are they? This is just going to be actual histology. Yeah. All right, so there are a couple great models in the lab that we have of a skeletal muscle cell that I actually wanted to go over. Unfortunately, I don't have them. Why won't you let me go back any further? There we go. But I will see if there's another instructor who does have pictures of them that I can use so we can spend some time of it, because it's a really cool view of the histology of the muscular system, but it doesn't look like they have it here. They may not have the model at CRC, may be one of the reasons why. But I will get that. But what I wanted to show you is, yeah, all along the sarcoplasmic reticulum, we're going to have it. So yes, the majority of them are going to be by the terminal cisternae, but they will run the entire length of the sarcoplasmic reticulum. Great question. Any others? All righty, excellent. Then what we'll do is go ahead and take our first break here. We'll take a 10-minute break. That means, so let's get back to the right place in the lecture slide. Oops, too far. There you go, perfect. All right, let's go ahead and take our first break. We'll take a 10-minute break. That means come back at 1:20, and at 1:20, we will then start the recording. All righty, see you in 10 minutes. All righty, any questions before we dive into the new material? All righty, then let's go ahead and pick up where we left off. We have now moved up to, again, our level of magnitude where we're talking about what is going on in a single muscle cell. And as we talked about, the first factor that determines how much tension, how much force a single muscle cell can produce is the amount of stretch to that muscle cell prior to the contraction, right? Again, if you think about it, the tension that is produced by a muscle cell, as we've talked about, is determined by the number of really two factors, right? So it's, again, we've combined it here into pivoting cross bridge forms. But let's, again, go with a simple drawing. Here's our Z-disc. Here's our actin. And then here in between it, we have our myosin. And of course, all of our myosin, to represent the heads, I'll just use arrows. That'll work. Great question about stretching, why it's so important. It is stretching, but not actually for this. We'll actually get to why stretching is so important before workout in just a minute. We talked about that a little bit, remember, when we talked about the skeletal system. Stretching warms up the joints, increases the amount of synovial fluid that is being produced. But there's more to it than that, and we'll talk about why it's important for muscles as well. It's actually a great question, and we will definitely get back to that. Let me see. Cheat and put that there. Okay, excellent. Make this one a little bit longer. So, as I mentioned, to generate tension, basically two things have to occur. The first is we must be able to form a cross bridge. And the second thing that has to happen is the myosin head must pivot. All right, seems simple enough. And again, really, to determine the tension, it can be something as simple as counting the number of times that can occur. Here we have a very simple myosin. This simple myosin has 8 myosin heads. And notice at this particular length of our muscle cell, only 1, 2, 3, 4 of the myosin heads could form cross bridges. So these 4 could grab, these 4 cannot. So in these 4 grab, there's plenty of room for them to be able to perform the power stroke and be able to pull. All right, this could get pulled this way, this could get pulled this way, and the result of that is we would produce, right, 4 myosin heads worth of tension, whatever that number would be. And again, the actual value of the tension isn't what is important. Okay, now let's take an extreme situation. I'm going to go ahead and erase those 2 there. What if we were to stretch the muscle out? And again, if you think of your forearm, there you are, and again, let's not make it you. There your best friend is, you guys are at the bar on Friday night, and when you're at the bar on Friday night, right, your buddies had 1 too many Long Island iced teas, and as a result of that, they gotten a little drunk and they've gotten a little rowdy. Of course, if it gets really bad, the bouncer has to come and remove them. And if you think about when a bouncer comes to remove someone who's being, ornery, typically they do one of 2 things. One of the things they'll do is they'll take the person's arm and they'll pull it behind their back. If you think about having your arm stretched out behind you, what you're doing is stretching the muscle cell. And when you stretch the muscle cell, you stretch the sarcomeres. And as you stretch the sarcomeres out, if you stretch the sarcomeres out far enough, lo and behold, we have this situation here where now none of the myosin heads overlap with the actin. And if none of the myosin heads overlap with the actin, can we form any cross bridges? No. And if we can't form any cross bridges, we can't pivot, and 0 tension is going to be produced. Notice if instead we shorten the muscle a little bit, remember the very first length, only 4 myosin heads could grab and pull. How many myosin heads can grab and pull now? Right, all of them, all 8 of them, absolutely. So this one is going to produce twice the tension that it did at the previous length. But here's the last thing. The other thing, again, remember there's 2 things. You have to be able to form the cross bridges, but you also have to be able to form the power strokes. Notice if we shorten the muscle cell enough, I'm going to have to cheat a little bit here and elongate my actin, where the actin interacts with each other, where the actin interacts with the myomesin. Now at this point, notice all the myosin heads are able to form cross bridges. But with the actin overlapping, with them intertangled with that myosin that's on the M line here, right? If we draw the myosin in here, are these myosin heads going to be able to form efficient power strokes? No. And so once again, the tension goes down. And if we think back to that bouncer at the bar, If they're trying to stop you, if you're being all ornery and trying to get into a fight, one of the things they'll do is they'll pull your arms behind you, stretching them out, decreasing the amount of force you can produce. Or the other thing they'll do is they'll bear hug you. When they bear hug you, basically they clench your arms up, right? Kind of like in a fetal position where your arm is all completely flexed. And if you're flexed like that, then the muscles are all shortened, our actin and myosin overlap. And again, the amount of tension you can produce is decreased. So notice the length of the muscle when we start determines how much tension you can produce. And you can actually experience this for yourself. What you can do is, and again, it helps if you have a partner, but you can also do this with a weight. What you can do is you can take your elbow and try to flex your elbow. And what you do is have someone put their hand against your wrist as a resistance. And you're going to do it in 3 states. You're going to start with your arm at a elbow at a 90-degree angle. We had a 90-degree angle. Yes, the tension is the force or is the strength, how much you're generating. At a 90-degree angle, try to contract your forearm and see how far you can do it. How hard or easy it is to resist their tension. Then fully extend your arm at 180 degrees and even hyperextend a little bit and see how easy it is to flex and against that force. And then also put your arm at 45 degrees. And when you put it at 45 degrees, try to flex. And as you'll see, the length of the muscle changes how much tension you can produce. We can actually measure this. I'm going to go ahead and clear my amazing picture here. And we can take a look at the pretty picture from your textbook. And here's the pretty picture from your textbook. Notice here up at the top, and let's go back to my spotlight, we have the optimal length. Notice this is the optimal length of our sarcomere, where there is lots of overlap between the myosin and the actin, but there's also lots of space for power strokes to take place. Notice as we stretch the muscle cell out, there is less and less overlap, and with less and less overlap, fewer myosin heads can grab and pull, and the tension goes down until you stretch the muscle cell out so much that there is no overlap with the myosin and the actin. And if there's no overlap of the myosin and the actin, then you are not going to produce any tension. Zero tension can be produced by that muscle cell. Conversely, as we shorten our muscle cell, right, again, there's less space to perform power strokes. So as there's less space to perform power strokes, we see a gradual decline in the tension. But notice here at point C right here, this is when the actin comes in contact with the myomesin. And now that it's come in contact and interfering with the myomesin, it's not as easy to pull the actin. And in fact, the actin can overlap entirely. So notice once we start messing up or getting in the way of other proteins, then notice there is a very rapid decrease in the tension that you are able to produce. All right. Notice our normal range of our muscles is typically in this highly efficient. And what's really cool is if you're in anatomical position, if you were to stand up and put yourself in anatomical position, most of the muscles in your body would actually be in this optimal range. Now, of course, the key word in that is most, and most means not all. There are some ones that are different, but most of the muscles of the body would be in optimal range. And again, you probably haven't thought about it in those terms, but you're aware of it because everybody hopefully at this point in this class has stood up in anatomical position at one point or another. If not, that's part of your homework before Thursday. Stand up and demonstrate anatomical position to somebody. But standing in an anatomical position is typically not how you stand waiting for the bus. As you stand waiting for the bus, right, we're in that body neutral position. And really the big difference between anatomical position and body neutral position is the position of your hands. In anatomical position, your palm faces forward, your palm faces anteriorly, and your thumb is lateral. In body neutral position, the way you stand towards the bus, your palm faces medially and your thumb faces forward. Your thumb is anterior. So that where your palms are facing your legs, that is that body neutral position. Right? But so when you're in anatomical position, most of your muscles in your body are at their optimal length to produce the most efficient amount of tension, most efficient amount of strength when they are contracting. All right. Questions on that? I think that one's a pretty easy one, right? How much room there is for power strokes and how many power strokes you have determine the strength. I think that one's pretty simple and straightforward. And we did all of that, so we did that. Excellent. However, the other factor that we talked about is this frequency of stimulation. All right, I have a couple times now today, and we will continue to talk about this, but as I mentioned, one, neural action potential, produces one muscle action potential, but we're going to add to that as well, because that one muscle action potential produces one twitch. So one neural action potential produces one muscle action potential, and one muscle action potential produces one twitch. Of course, now we have a new word, a new term, so we have to define that new term. A twitch is a single stimulus, contraction, and relaxation sequence in a muscle cell. Here, we see some pretty examples of some different twitches in different muscles of your body. Notice different muscles of your body have different speeds for their twitch. Notice all three of these muscle cells are all getting excited at the exact same time. However, here, one of your eye muscle cells, and again, by eye muscle cells here, I don't mean the ones inside your eyes, I mean the ones that move your eyes in space. So if you can look up right now and look down right now and look left and right, if you can cross your eyes in the middle, that movement of your eyes, those are the eye muscles. And notice they have a very short twitch. They go about 15 milliseconds. Right here in red, we have your gastrocnemius. Again, in fairness, we haven't talked about the leg muscles yet, but hopefully you have started looking again. Your gastrocnemius is a nice superficial muscle of the leg. In fact, it is what we commonly refer to as the calf muscle. So as you look down at your calf muscle, right, or admire your significant other's calf muscle, what you're really admiring is their gastrocnemius. And look at the twitch for that, from stimulus, takes about 40 milliseconds. And then deep to your gastrocnemius, one of the important stabilizing muscles that allows you to stand for hours and hours on end is your soleus. Again, notice we stimulate it at the same time, and we get a twitch that occurs, and that twitch lasts over 100 milliseconds. So notice the timing of all three of these are different. But the one thing that is consistent between them is they all have three distinct phases, right? As we talked about in the twitch, there is a stimulus, there is a contraction, and there is a relaxation. We'll talk about these stages in detail, but I want to at least identify them now. And again, we can look at them and think of these stages in terms, when we're identifying them, think in terms of the amount of tension that is produced. Again, tension is the physics term or the force that these things are producing. And notice even your textbook doesn't bother to give us a value for that tension. It just shows that there's from an increasing tension as these go. And that's the key. In stage one, notice, and again, we'll give names to these in a second. Actually, I'll go ahead and write this out now. Can you guys see my eye bar if I do this? I want to be able to continue to type here, but I don't want to use this. Can you guys see the eye bar when I move it around? Okay, excellent. Notice all three of these are stimulated at the same time at this 0 point on the axis here, the x-axis. But notice it doesn't start producing tension right away. There's a little bit of delay before tension is produced. So the first stage is what we call the latent stage, or latent phase. And notice during this stage, no tension is produced. The second stage is this part where we have an increase. Notice that increase is slower in the soleus, a little faster in the gastrocnemius, and really fast in the eye muscle. But in all of those, as we said, we have a contraction phase. And during this contraction phase, tension increases. Lastly, we have the relaxation phase. And again, I think this is one of those things that is confusing for someone. When we say relaxation, we do not mean when the cell is back at rest. The relaxation phase is basically from when you go from the peak back down to rest. So notice again, our eye muscle had a very fast contraction phase, and it has a very fast relaxation phase. Our gastrocnemius, a slightly slower contraction phase, a slightly slower relaxation phase, and our soleus has a nice slow contraction phase and a nice slow relaxation phase. So during the relaxation phase, it's not when it's back at rest, but it is when the tension decreases. Oops. So again, we're going to identify all the stages, talk about everything that is going on during those stages, but the real key difference in identifying these stages is to do it and identify them from how much tension there is being produced. So during our latent phase, there is no tension being produced. During the contraction phase, we are having an increase in tension. And during the relaxation phase, we are having a decrease in tension. Alrighty, we are going to pluck one of these twitches out to take a look at it independently. And since the gastrocnemius is the one in the middle, notice we have that here. And notice here, again, phase, period, same thing. These terms are interchangeable. We have those here, and we see it in our single twitch. We are looking at a single twitch now. Now, there is a couple things. Let me see. If I take this and move this here. Sorry, I have to play with what I'm doing, and let's of course clear all of these things. Notice again we have our y and our x-axis here. There is some unique things that I want to point out and emphasize to you here. Notice again they haven't given us a value for tension, but clearly down here tension is going to be 0. And then up here is whatever number of the joules or whatever it is that they're using to measure it. We don't care about that. That's too much of a physics thing. And we expect the 0 point to be right here at the cross point of our x and y axis. Well, let's talk about Charley horses and random twitches like in your eye in a second. Let's understand how it works properly first, and then absolutely we can talk about those irregular twitches that we get. So let's talk about the normal one first, and then we can kind of make sense of these. All right. What I want to point out to you is that normally the other 0 would be here. But notice our 0 is way offset. And that's because our 0 point for this is, notice when it receives its stimulus, or more specifically, this is when we produce the muscle action potential. So stimulus is basically when we produce our muscle action potential. And so if you think of, oh, what happened there? I don't know why that went away. Interesting. All right, let's do that again. That is so weird. All right, I don't care anymore. All right, I care a little bit. Oh, there's an extra space in there for some reason. All right. All right, whatever. There, we produce our muscle action potential. That is our 0 point. If you think about the process that we just all started with, when we go to contract a muscle, the first thing that happens isn't our muscle action potential. If you think about it, the first thing that happens, this point right here, is when we make the decision. Then once we make a decision, we produce a neural action potential. That neural action potential spreads down the axon to our synaptic end bulb. When it gets to the synaptic end bulb, it opens voltage-gated calcium channels, yammer, 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 everything that goes along with that. So if you think about it, this part here, all of this before 0 is everything that happens before we produce our muscle action potential. So if you think about it, all of this is our communication at the neuromuscular junction. Right? That is what is happening in this period here. And notice that's not a part of the twitch because that's not what's happening on the muscle cell. What is happening on the muscle cell starts right here at the 0 point. Oops. Right here at our 0 point, and at this point right here, this is when we now produce our muscle action potential, and that is our beginning of our twitch. But notice the first 3 or 4 milliseconds of this, we have this latent period. As we talked about, the primary way we identify the latent period is that there is no tension produced. And again, if you think about what we know about how a muscle contracts, you know that as soon as that muscle action potential is produced, myosin isn't suddenly pulling on actin. You know that first we have to spread that muscle action potential down the sarcolemma, down the transverse tubule, depolarizing the triad, opening up those voltage-gated calcium channels, using that calcium to move the regulatory proteins. So if you think about it, during this latent period, this is when we are moving the regulatory proteins. Right? It's our excitation contraction coupling if you think about what's happening during that latent period. So before we move the regulatory proteins, we can't produce any tension. And so there's a brief period of time. Notice it doesn't take long. It doesn't take long to spread that action potential down the cell, to depolarize the triad, to open those calcium channels and let the calcium out and start moving the regulatory proteins. But there is a brief period of time, a few milliseconds, and that is the latent period. Now, when we send that action potential down and we open up those voltage-gated calcium channels, does all of the calcium come out of the sarcoplasmic reticulum all at once? No, it's going to be a bit of a jailbreak, a little bit at a time. And so what's going to happen is the first 3 calcium come out, and they bind to 3 troponins, and they move 3 tropomyosins, and 3 myosin heads can grab and pull. And if 3 myosin heads can grab and pull, are you going to produce a massive amount of tension in your muscle cell? No, exactly. But then 10 more calcium come out, moving 10 regulatory proteins. And then 45 more calcium come out. And then 352 more calcium come out. So notice during our contraction phase, there are basically 2 things that are happening. There's going to be an increase in the amount of calcium that is released from the sarcoplasmic reticulum. And as more calcium is being released, that means we're able to move more regulatory proteins. And if we are able to move more regulatory proteins, more myosin heads can grab and pull. And if more myosin heads can grab and pull, then the amount of tension we produce increases, which is, of course, our goal of the contractant phase. So notice we see this continuous increase in the amount of tension that is being produced because more and more calcium is being released. And as more and more calcium is being released, we're able to move more and more regulatory proteins. And the more regulatory proteins we move, the more myosin heads can grab and pull, and the more tension we produce. Of course, remember, this was produced by one muscle action potential. And maybe we need to put that up at the top. To remind ourselves of that. These muscle action potentials are big positive signals. But as we've also talked about, the muscle action potential doesn't last for a very long time. One muscle action potential is very short. Right, a couple milliseconds. What that means is that while we were able to open up all those voltage-gated calcium channels and a lot of calcium was able to come out, the cell repolarizes very quickly. And of course, as we know, when the cell repolarizes very quickly, our voltage-gated calcium channels close, and those pesky pumps put the calcium away. Now, when those pumps are putting the calcium away, are they able to grab all the calcium at once and put it all away all at one time? Again, no, absolutely not. So what happens is our relaxation phase. During the relaxation phase, what happens is more and more calcium is put away. So notice what's going to happen is we are going to get a decrease in the amount of calcium that is now available in the cytosol. Again, it doesn't all get put away at once, but more and more of it is getting put away, and as more and more of it is getting put away, less and less is available in the cytosol. If there's less calcium in the cytosol, that means that with less calcium, there's less calcium to bind to the troponin. That means fewer regulatory proteins are moved. If fewer regulatory proteins are moved, that means fewer myosin heads can form cross bridges. And if fewer myosin heads can form cross bridges, then the amount of tension we produce, decreases. And that is the relaxation phase. Until the tension equals 0. And when the tension equals 0, the cell is back at rest. And so that's what we're seeing here with a single twitch. We produce one muscle action potential. That one muscle action potential produces one twitch, right? And let's emphasize that. Here, we'll change colors of that to make sure. Whoops, oh, I didn't mean to do that, but that's fine too. So that one muscle action potential produces one twitch. And that twitch has 3 phases to it. Phase 1, which is the latent period, this brief period of time where from when we stimulate and generate that muscle action potential no tension is produced. And that's basically because we have to spread that muscle action potential, open up those voltage-gated calcium channels, which is essentially what's happening in the excitation contraction coupling. Of course, we don't allow all the calcium out at once. At first, only a little bit of calcium comes out, but then more and more and more and more. And so as more calcium is released, we hit our second phase of the twitch, the contraction phase, where we have an increase in the available calcium. So more regulatory proteins are moved. So more myosin heads can grab and pull. So more tension is produced. And that is the key to the contractile phase. In the contractile phase, tension is going up. Right? Latent phase, tension stays the same, does not change. Well, let's talk about the other twitch in a second. We'll get to the other twitch. That's a great question. That is absolutely the next question we're going to get to. That's an awesome question. But let's finish this twitch first. Absolutely. So we reach that. But again, it's going to peak pretty quickly. Right? We're talking about milliseconds here because that muscle action potential only lasts for a short period of time. And since it only lasts for a short period of time, very quickly, the cell repolarizes. Our voltage-gated calcium channels close, and our pumps start putting the calcium away. And when our pumps put the calcium away, we have a decrease in the available calcium. We have a decrease in the number of regulatory proteins we can move. We have a decrease in the number of myosin heads that can grab and pull. And so our tension goes down until it reaches zero. Now, you guys have asked two great questions, so let's go back and answer those real quickly. This is a twitch. And again, it occurs when a muscle cell produces a muscle action potential. Usually, that is when I make the decision to stimulate a muscle action potential. But maybe my muscles are fatigued from working out, or maybe my eye muscles are tired from staring at the screen for 4 hours while your instructor lectures to you, or those types of things. When there's a fatigue or there's imbalances in ions or irritation or injury or things like that, can you get uncontrolled, isolated action potentials that are produced that cause a rapid twitching of the motion, like that eye twitch that you can get when you're tired or fatigued or something along that? Absolutely. Again, is that how we want to normally move our body through space? When you come to lift that coffee towards your face, right, the first thing in the morning, you make that scalding hot cup of coffee. Do you twitch the muscle? Your arm jerks in 40 milliseconds to bring the coffee up and then splashes the coffee all over your face? No, absolutely not. If we are going to want to produce a sustained contraction, and also too, if you think about it, that one twitch, when the muscle in your arm or the muscle in your leg twitches, it's typically not enough to jerk your leg or jerk your arm through space. You just see the muscle generating the tiniest bout of tension from that one little twitch that is being produced. So, as was pointed out by Gabriella, we need to produce more than one twitch to move our body through space. Now, when it comes to moving our body through space, though, again, if I'm lifting my coffee cup, I don't want to produce one twitch and then a second and then a third and then a fourth and then a fifth, and every time wait for it to go back to rest before I do that, because then my arm's jerking all over the place, and that's going to be a problem if I'm having a cup of coffee. So absolutely, we are going to be able to produce a second twitch before it reaches the resting phase. In fact, we're going to want to do that, and that's the way we normally move our body through space. However, and here's the key. I'll get to that second question in just a minute. Here is the key. We can absolutely produce a second twitch before the first reaches the resting phase, but there is a limit. And that's why, okay, I want to make sure any questions on this. Oh, I forgot to put my 3. Let's go back here and put my 3 for the 3 stages of a twitch. Because if I ask you to define a twitch, then this would be your essay question. You need to tell me that this is a twitch. I maybe have to draw it, although I don't have any idea how we would draw this online. I don't even know if that's possible. But at least describe the phases, identifying the phases, talk about what's happening during the phases. This is how you produce a single twitch. And a single twitch is one neural action potential, produces one muscle action potential, produces one twitch. Now, I would not go so far as to say a twitch is a muscle contraction, although it is causing a change in tension in the muscle. We'll talk about contractions in just a minute. We want to be careful with that word and with definitions. But what we are doing is producing tension within a single muscle cell. And that is what we are doing, producing tension. All right? Not the same kind of twitches that has when the body falls asleep. Those are typically more powerful twitches, but they are a more coordinated type of twitches. But what I do want to come back to is, again, that first question about can it be produced before the first twitch reaches its resting phase? The answer to that is absolutely yes. However, this is a single twitch, and we want to produce a second twitch, and that's the key. When we talk about a single twitch, everything that we've written here on the screen describes and talks about this single twitch. However, there is going to be a time, and let's emphasize this by changing the color, where we want to produce a second twitch. All right? When we want to produce a second twitch, there is a limit on how fast we can do this. So again, what I'm going to talk about right now has absolutely nothing to do with the twitch that is on the screen right now. What I am talking about solely is how we produce a second twitch. So if we're going to want to produce a second twitch, there's a limit on how fast we can do this. That limit is called the refractory period. Basically, if you remember back to our first lecture on the muscular system, one of the things we talked about is how muscle cells are irritable. Irritable, remember, means that they receive a stimulus and respond to it. Well, one of the interesting things about muscle cells, and when we get to our neurons, we'll see they do the exact same thing. When stimulated for a very brief period of time, our muscle cell loses its irritability. Meaning that it cannot be stimulated again. Doesn't matter what you do to it. You hit it with a bolt of lightning, you plug it into a light socket, it will not produce a second action potential. The good news is this period of time is very short. It's only about, let's say, let's go with a number 1 to 5 milliseconds. Let's go with the number 5 milliseconds. I like that better. It's only about 5 milliseconds. Notice by 5 milliseconds, the cell here is barely starting to enter its contractile phase. All right, so it's barely starting to be able to produce tension. However, in this brief period of time right here, no matter what you do to it, you cannot get it to produce another action potential. And if you can't get another action potential, you can't get another twitch. So there's a big, huge, solid line right here where no matter what we do, we cannot get the cell to fire a second twitch. However, after this point, after that refractory period, and all the way through to its rest and beyond, we can produce a second twitch. So there is a very brief period of time known as the refractory period. No matter what we do, we cannot get it to fire a second action potential. It loses its irritability. It no longer can be stimulated. But after that period of time, it can be stimulated together to produce a second twitch. In fact, that's what we're going to have to do to move our body through space, because we're not going to rely on just one twitch to move our body through space. All right, so any questions on our twitch, the first twitch, or this concept of refractory period, which affects our ability to produce a second one? Any questions on that before we move forward? All right, I've written it all out here. I've got some pretty pictures on your slide to help us to remember these things. So again, notice as you look here, let's move that out of the way for me. Our latent period, again, this latent period is when our action potential, muscle action potential, spreading down the sarcolemma, spreading down the transverse tubules, all right, spreading to the triad, opening those voltage-gated calcium channels. Then calcium starts coming out a little at a time, and then more and more and more and more. And again, during the contraction phase, the amount of tension increases. More calcium is out, more regulatory proteins are moved, more myosin and actin pull, more tension is produced. Lastly, our relaxation period, the amount of tension decreases because, again, calcium is put away. Less calcium is available, fewer regulatory proteins can move, fewer myosin heads can grab and pull, and so our tension goes down until we reach rest. Once we reach rest, then tension is back to zero again. So those are our 3 phases of a twitch. And like we said, the refractory period has nothing to do with this twitch. It does not affect this twitch at all. This twitch on the board, the refractory period means nothing to it. What the refractory period means is when can we produce a second twitch. And remember, we have to basically wait till that twitch reaches that end of that refractory period, and then we could produce another twitch again that we could add to the previous one. All right, clear by drawings. Questions on that? Now, we understand the concept of a twitch, but like I said, it isn't necessarily useful by itself. But there are some cool concepts, some more, and let's again write this out. Characteristics of our muscle cell. And one of those characteristics of our muscle cell is the concept of TREPI. TREPI is a really cool concept. And this will get back to actually one of the questions that was asked earlier. Let's go ahead and draw our graph again. So here is our graph. Of course, we have to label our graph. This is tension. Over here, this is time. Oops. Down here. And as we talked about, if we stimulate, so here, bing, at this point, we get, and I'm just going to draw an M there for the muscle action potential. When that muscle action potential occurs, we produce a twitch. And I'm going to purposely exaggerate the timing of this so that we get it. There is a latent period. There is a contractile period. There is a relaxation period. All right, 3 stages to that twitch. Stage 1, stage 2, and stage 3. All right, we comfortable with that? Now, if I wait 5 minutes, and after 5 minutes, I stimulate it again, I produce another muscle action potential. If I produce another muscle action potential after 5 minutes, what am I going to get as a result of that? Well, 1 muscle action potential gives me what? Apparently, you've all fallen asleep. I understand. Come on, give it to me. 1 muscle action potential produces what? 1 twitch. There you go, absolutely. And guess what? If we had waited 5 minutes, and again, this is where we end up dealing with the limitations of my drawing skills, but absolutely, Ashley is correct. What we are going to get is we are going to get a second twitch, And this second twitch is, and I'm going to cheat and do it this way, will have the exact same tension. I know, well, my drawing skills aren't quite good enough, but let's cheat and do it that way. They will have the exact same amplitude to it. The exact same amount of tension is produced. It's going to be identical. And I'll cheat by writing that. All right, so after 5 minutes, we would end up with an identical twitch to the one we had before, with the exact same amount of tension. But here's part of the cool thing. We don't actually have to wait 5 minutes before we stimulate it again. What we can do is, and let's draw another line. Oh, wait, hold on. Yeah, actually, that'll work. All right. This is the point where our cell reached rest. And at that point where our cell reached rest, we can produce another muscle action potential. So our cell has reached rest, and immediately after it reaches rest, we stimulate it again. And if we stimulate it again, what's going to happen is we are going to get another twitch, and that twitch is going to have a latent period, and it's going to have a contractile phase, and it's going to have a relaxation phase. And this time, this wasn't an error in my drawing skills. The result of this is we produce more tension. So the result of the second twitch is bigger. We let the tension equal 0 again. So again, at this point, and I'm going to cheat and put it up here for more space, our tension is equal to 0. At this point, we stimulate it again. And when we stimulate it again, what happens is, again, we are going to produce a new twitch. And this new twitch is going to be bigger than the last. This situation where we have increasing tension is a characteristic of our muscle cell called TREPI. Yep, so, well, so that's the key, right? And that is the key to TREPI. Notice with TREPI, we are not actually building tension on the first twitch. And that's really the key to this. With TREPI, what happens is, again, here's the key to it. We let the cell reach rest. If we let the cell reach rest, remember what that means is tension is equal to 0. Or tension equals 0. And so if that's the case, we're not adding tension to what is there. Because what is there is 0. But what we are doing is we immediately stimulate it once it reaches rest. And if we do that, oops. If we do that, when we do that, we produce more tension. That is truly what our definition of TREPI is. TREPI is this concept, this characteristic of a muscle cell. When you let its tension reach 0, so we're not adding to what was there before. We're not adding to what was there before, but we still get a bigger twitch. So the real question becomes, why the heck does this occur? What is going on here that is causing this weird effect? Well, here's the key. Here's the answer to that. Let's think about this. We have in our first twitch, we have an increase in tension of our twitch because remember, there's an increase in calcium. More calcium is being released from the sarcoplasmic reticulum, and the more calcium is released, the more regulatory proteins we're able to remove. More regulatory proteins we move, more myosin heads can grab and pull, more tension. The relaxation phase is when there is a decrease in calcium. The calcium is getting put away, and as the calcium is being put away, fewer regulatory proteins can be moved, and as fewer regulatory proteins can be moved, then again, less tension is produced. So here's my question. Right here, when our tension reached 0, do you think all the calcium has been put away? No, as it turns out, it hasn't. And that's really the key. What happens is there is residual calcium. When the tension equals 0, there is still residual calcium. And that residual calcium is what gives us effect. Notice, if we waited 5 minutes, after 5 minutes, all of the residual calcium got put away. So our second twitch after 5 minutes was the exact same size as our first. I know this concept can be a little tricky to understand, so let's use really simple numbers. If we use really simple numbers, let's say that when we produce a muscle action potential, the cell releases, oh, I don't know, let's go with a good number. Let me get a color first. 100 calcium. Oops. 100 calcium is released when we produce a muscle action potential. All right? However, when our tension reaches 0, and for this I need to get rid of this one, only 90 has been put away. So of course, how many is just left? There you go, 10. Now we produce a second muscle action potential. And when we produce a second muscle action potential, as we just talked about, a muscle action potential produces 100 calcium. But remember, there's still 10 left. And so now we have 110 calcium that is available here. 110 calcium can move more regulatory proteins than 100 can, and we get more tension. Notice when during the relaxation phase, again, 90 is going to be put away. But now notice there's 20 left. So when we stimulate it again, there is now 100 calcium released plus the 20 left over. So, of course, now we have 120 calcium to move regulatory proteins, and more regulatory proteins can be moved, more myosin heads can grab and pull, and more tension is produced.
Hello.
Yes. Was that hello just to say hi, or was there a question? I guess it was just say hello. Well, then hello back. Excellent. So notice this concept of TREPI has to do with residual calcium. There is going to be residual calcium left in the muscle cell, even when it goes back to rest. And so if we stimulate it again, oh, no problem. If we stimulate it again, then we are going to get more tension produced. This goes back to what someone was talking about at the beginning of class when we were talking about warming up before an exercise, doing that stretching before an exercise. And probably the greatest example, which of course we now have to wait an extra year for, is the Olympics. My favorite example of the importance of TREPI is always in those sprinters. Usain Bolt, when he is going for another world record in the 100-yard dash or 100-meter dash. If you think about those sprinters, sprinting for 100 meters requires a tremendous amount of energy, right? And so when they're going to do that 100-meter dash, they aren't sitting there totally still, conserving all of their energy so that they're ready to run. They're constantly in motion. They're jumping up and down, they're bouncing around, they're shaking their arms, they're shaking their legs. They're constantly moving, even as they're getting into the blocks, right? For the start, they're moving as much as they possibly can into that last possible second, because what they're doing by moving those muscles, they're producing residual calcium in that muscle. So that with that additional calcium, when they make that burst out of the block, that residual calcium allows them to produce a more powerful contraction, and they are faster out of the block as a result of that. So warming up the muscles absolutely is a real thing. Warming up those muscles gets that residual calcium in there and allows for a more powerful initial contraction. And so that's why they're warming up. That's why they're constantly in motion before that. It's because of this characteristic of our muscle cell called TREPI. All right. Questions on that? All right, excellent. So hopefully this makes some semblance of sense. Now, there is going to be a limit of how much you can get by TREPI. Because again, if you think about it, if there's too much residual calcium, then the muscle is just going to continue to continue to contract. So there is a peak. And if I like this picture, so let me go ahead and I'm going to steal that screenshot of that. But I'm going to go ahead and erase it now. I'll post this on our Canvas site like I did the other ones. Let's clear this. And we can look at the pretty pictures from your textbook that show this same concept. So again, here's the key. For a TREPI to occur, it has to be stimulated. The muscle cell must be stimulated immediately after the relaxation phase. Again, the key here is the tension equals 0. We are not adding tension to tension that is already there. The tension equals 0. But if we do that, even though the tension is 0, our second stimulus is enhanced. Here, we have the pretty picture from your textbook, similar to the one that I showed. There's our first twitch. It gets 0, we stimulate it again, and it gets bigger. 0, we stimulate it again. 0, we stimulate it again, and it gets bigger until eventually it reaches a maximal tension in TREPI, whatever that tension is, that limited tension. So again, notice we're able to do this, and sometimes up to 30, 50 times, slightly bigger, slightly bigger, but we are eventually going to reach a max. This is important. This is useful. But again, this isn't the way we're going to move the body through space. Because if you think about trying to lift that coffee cup to your lip, you don't just do a small twitch that barely moves your thumb, and then a slightly larger twitch and a slightly larger twitch. And you don't keep twitch, 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 twitch of your arm until finally that coffee cup reaches your mouth. So this is useful, and it's an important characteristic of the muscle cell. But here's the other thing. Notice the key to TREPI is we have to let the tension go back to 0. And if we go back to 0, we can't add tension to what's already there. However, as was already asked and we mentioned earlier, we don't have to wait for tension to reach 0 for us to twitch it a second time. And if we don't wait till that twitch reaches 0, then that means we can actually add twitches together. And that's our second important characteristic of the muscle cell, this concept of wave summation. The key with wave summation, again, as we just mentioned, is we want to add twitches together, right? It's the opposite of the last one. We want a second twitch before the first goes away. Right? We want to add twitches together. We want to stimulate this a second twitch before the first twitch goes away, before the relaxation phase. Because if twitch reaches 0, then there's nothing to add anymore. right? And that's not what we want. We want to be able to add. Now, as we also mentioned, whoops, there is an upper limit to how quickly we can stimulate it. So obviously, we have to do it before tension reaches 0, but we have to do it after the refractory period. As long as we do it between these two, then what we can do is we can add the twitches together. And notice that's what's happening here. On this illustration that we see, we have our first twitch that comes up and would come down. But we stimulated a second time before it's had a chance to go fully down, and we produce a second twitch, and we add that second twitch to the first. And then we stimulate it again, and we can add a third to the first two. And then we stimulate it again, and we can add a fourth. If we compare this to trepi, right, our trepi on this, just by trepi alone, would probably be somewhere within this range. So notice with wave summation, we are able to generate much more tension. Than with trepi alone. And so we can generate massive amounts of tension in this muscle cell to be able to move our body through space. And this is how we normally move our body through space. We use wave summation. Now, is there a limit to how high we can produce it? Absolutely. And in most cases, we are not actually going to reach that maximum. There is a maximal tension. Notice as we produce these wave summations, we add the waves together. Typically what happens is we reach this point where we have an undulating peak near the maximal tension, but doesn't actually reach it. This level of undulating tension near the peak is what we call an unfused tetanus. And this right here is the normal way we contract our muscles. Right? Now, if you think about it, that doesn't make a lot of sense because we'll get to the max in a second, as you're lifting the coffee towards your mouth, is the coffee cup shaking while you're doing that? No, it's typically very smooth in that motion. But the reason for that is even as light as your coffee cup is, you're not going to rely on just one muscle cell to move it, right? It's like a choir. I don't know what your singing voices are like, but again, if it's good enough for Elton John, it's good enough for us. So I don't know if I told you guys yet, but tomorrow night, our 4:30 class, we are going to be presenting a Zoom broadcast to the world where we're going to be singing as a choir. All right? And hopefully a handful of you would be willing to do that. But obviously, if we all did it together, if all 30 of us sang together, that probably wouldn't be that horrible. Our voices would all kind of meld together. And, you know, most people, especially if it's maybe 30, but even in a choir of 200, most people don't have a problem singing in a choir of 200 because your voice is going to blend in with everybody else's. However, many of you would probably have major problems talking or singing solo or even in a duet. When we're lifting even something as light as that coffee mug, we're using lots of muscle cells. Every single one of those individual muscle cells is undulating in its pattern, but they're all undulating at different times. And that is why it smooths out and we get a smooth, continuous contraction. However, where you can see this undulation is in two instances. One, when you are near the peak. So if instead of lifting a coffee cup, you had to lift your chest of drawers in your bedroom, right? How long could you hold up your chest of drawers for? Right? Or you're helping a friend move a couch. and they're having trouble getting their keys out. So there you are standing outside their door, they're fumbling with your keys, and you're trying to hold that couch up. And as you're trying to hold the couch up, notice your arms start to shake a little bit. The reason they're shaking a little bit is because you are reaching the maximal strength you can generate. And so what's having to happen is all the muscle cells are all firing together to try to produce enough tension to hold it up. And when they're all firing at the same time, that's when you start to see that undulation. The other time you see it is when you're only using a few muscles. Lifting that coffee cup, you do that in a nice smooth function. But how easy is it to thread a needle? When you're trying to thread that needle, often your hand will shake as you're trying to thread that needle. And the reason your hand shakes when you're trying to thread that needle is because you are firing just a few muscle cells. And when, again, you're singing in a duet or in a trio, those individual voices stand out more. So when you're only using a few muscle cells to do that fine discrimination, like trying to thread a needle, then you tend to see those undulations more. Now, the question was, and it's a great question, what about bodybuilders? Do they reach the max? They can, although usually they don't. However, are there instances where we can reach the max? Absolutely. There are definitely those instances, all right? We get that sustained wavering contraction, when it is technically possible to get a muscle to contract at its peak. And when it contracts at its peak, we get a smooth, sustained contraction called a complete tetanus. Typically, we do not want to contract a muscle at a complete tetanus because there the muscle is producing its maximal amount of tension. And at that point, the muscle is much more likely to be damaged. We're much more likely to damage this. So again, bodybuilders, when they're doing their big, bulky exercises to build up for a competition, typically won't use this. However, if they're going for the world record in the, you know, the grab and jerk or whatever it is, or some bench press or something along those lines, when they're trying to go for the world record of those things, can they sometimes do that? Can little old ladies lift up cars and flip them off of little babies that are trapped underneath sometimes? Sure, absolutely. If you were to, because you're wandering around the wilderness now because you're not allowed out, you're hiding and sneaking, trying to go over to a friend's house, and the police are trying to stop you from getting outside of the house, and you grab a hold of that electrified fence, and that electrical current goes through your arms. Well, all your muscles in your arm then all contracted that complete tetanus? Absolutely, right? And lock up. In fact, all of them will. So it is possible to produce at a complete tetanus, but it can have more risk of damage. So it's typically not going to be the normal way we're going to want to move our body through space. What happens is you're either flexing at the max because, again, you're trying to generate a massive amount of strength, or they can, especially for the dexterous activities, they can be trained for that. When I was in neuroscience, when I was in graduate school and for my neuroscience degree, one of the things that we would do to practice for our neurosurgeries is our professor would have us using a pair of forceps. And what we had to do is we had to tie a string around a single die inside of a jar. So she would put a single die into a jar. You then had to, with a piece of string and two forceps, reach in there and tie the string around it. Like I said, your hand may or may not wobble as you're trying to thread that needle, but if you do a lot of sewing, your hand is a lot less likely to wobble as someone who's never threaded a needle before, because you can learn and smooth out those behaviors by doing that. So again, you can train the muscles to do that, but our muscles typically don't want to do that. So it's either going to be something artificial, extreme stress, or again, extreme training. You can get to that complete tetanus. But it's not the way we typically want to move the body through space, because when you're at a complete tetanus, you're much more likely to damage the muscle. All righty. Questions on that? All right. That is everything that I wanted to cover for a lecture standpoint today. But we do need to still spend a little bit of time. I want to talk some more about some of the muscles, some of the wonky muscles. And as we move from the core of the body, the trunk of the body, to the arm, we need to talk more about these multi-headed and that whole magic term, be specific, and things along those lines. So any questions on these concepts? Again, notice we're still at this point, we are still talking about a single muscle cell. So we have talked about how we produce tension in a single muscle cell and how we can modify that tension in a single muscle cell. So any questions on these concepts in a single muscle cell before we put our lecture away for the day? All right, so let's go ahead and take one more break. We'll go ahead and make it another 10-minute break, and then we'll come back during this break. If you have not done so already, grab your red and blue pens, get your muscle list, get your origins and insertion list, and again, grab an atlas or a textbook or something like that so you can look at the pretty pictures while we're doing this. And so we'll come back at 2:45, and at 2:45, we will pick up from here. So I'm gonna move that down out of the way. Get rid of that. And then here. So again, we'll come back at 2:45 start. And then remind myself to start the recording. All right, see you in 10 minutes. Let's switch gears into the lab mode. Let's see, where do I need to go for that? And that, close that, start this. Perfect. Alrighty. Alrighty. So where we are at now is we were working our way through the muscles that move the trunk of the body. Let me get all my chat thing and participation thing back up and all those fun things so we all can see that, or I can all see that. Oh, and that's the other thing I needed. Oops. Hold on, I gotta stop sharing for a second. Minimize all that. There we go. That's the other thing I need. Perfect. All right. So as I mentioned, we were working our way through the muscles of our core of our body. And let me bring up the last thing I need to get is my study list. We had gone through some of the muscles, but what I want to leave us with, as we talked about last time at the end, I want to talk about the intercostals and the abdominal muscles. Because there is some similarity on there that I think will help us to appreciate and understand them. So what we see here, and I need my annotations, are the intercostal muscles. As the term intercostal indicates, these are the muscles that are located between the ribs. Right, so here between the ribs, we can see this, and there are two sets of muscles between the ribs. I think if we actually go to the whiteboard, we can kind of do a real simplistic view of this. So let's go ahead and clear all of this information from before and do some simple drawing. Here is the rib on one side. Oh, actually, let's move this down. And move this down. And then we'll draw two more on the other side. That works. we'll cheat and move these a little bit. So move this to the side, move that to the side, try to line them up a little bit. And then, of course, in the middle, we have our sternum. And as we also know, when I can cheat and use my highlighter, we know that there is going to be cartilage that connects these true ribs directly to the sternum. So there's my cartilage that we can emphasize with that. And that, in a most sophisticated kindergarten sense, is what our rib cage looks like. At least from a front view. Now, as I mentioned, there are two main types of muscles here found between the ribs. They are the internal intercostal and the external. And there are three primary ways that our internal and external intercostals are different from each other. As the name indicates, our external intercostals are going to be superficial. They're external, they're on the outside, whereas our internal intercostals are going to be deep. They're closer to the inside. A second way that our intercostals are different, and did I start the recording? Yep, excellent, okay. A second way that our intercostals are different is that our internal intercostals are more medial. Oops, where is that? Not ready, there we go. They are more towards the sternum. In fact, they actually touch the sternum. And the cartilage. Whereas our external intercostals are more lateral and typically do not touch the cartilage. Lastly, the internal and external intercostals are different in their orientation. As we know, muscles are made up of fascicles that are lined up. Both the internal and the external intercostals are parallel muscles. And in the case of our external intercostals, our fascicles project, let's say it that way, down, oops, and in, whereas in our internal intercostals, project down and out. So what this means, if we're going to draw them, and our internal intercostals, we will use, we'll go ahead and use purple for these. The internal intercostals, again, they start at the midline, their fascicles go down. No, that's not purple. There we go. So let's undo that. Their fascicles go down and out. They start more medial, whoops, meaning they start at the rib cage, the sternum, I'm sorry, and they work their way laterally, and they project down and out. Notice they do this on both sides. So on this side as well, they are going to project down and out as well. Yes, so we'll get to that. Their internal are deep to the external, so we're going to see that in a second. That's, again, we have to see that overlap to do that. Let me just draw a couple more of these, and we can get that. So those would be our internal intercostals. Our external intercostals, and let me get that list back up here as well. Sorry, I'm trying to draw and juggle these all these windows at the same time is tricky. So we want to be off of that, and we'll go with green for this one. So we'll make our internal intercostals green. So our external intercostals are going to be green. And if we draw those, they are lateral. Their fascicles come down and in. And again, these are superficial. Oops. So these go over the top and will cover and obscure. And we can cheat by using the highlighter to emphasize that. They're going to cover and obscure the internal because, again, they are superficial. The same thing is happening on the other side as well. Again, external intercostals, fascicles come down and in. They overlap the internals. They're more lateral. They don't make it to the cartilage. And so there are those three big differences in their anatomy. All right. We have a pretty picture from your textbook that does a good job of showing this. So let's go ahead and go back to our picture, because here we see them individually. But like I said, what I love about your textbook is it does a really nice job of pulling the muscles out and showing them independently. So notice here we have, I keep losing my annotation thing. The internal intercostals, notice they are medial. We can't tell they're deep because they've been pulled out by themselves, but we can see they're medial. They go to the sternum, they go across the cartilage, and their fascicles are coming down and out. Here's our externals. They're more lateral. They don't make it to the cartilage. They don't make it to the sternum, and their fascicles come down and in. And if we go to the previous picture, we can see them overlapping with each other. Notice here we see the externals coming down and in are superficial, and the internal intercostals are deep behind them. So that's that third characteristic. Notice they did cheat a little bit. They do have this one touching the cartilage. Again, artistic license. They typically don't come this far forward. I think they just wanted to give us a better view of them overlapping. So they have shown it this way. But typically they do not come out to the cartilage. They stop before the cartilage. So anatomically, they are very easy to distinguish. And what's also cool about them is they are also different in their function as well. Both of these are part of what we call those accessory respiratory muscles, right? So these are accessory whose job it's going to be to help us with we want to take an exaggerated breath. As we talked about, while you're sitting here calmly just listening to me talk, you're primarily just using the diaphragm. The diaphragm is your primary muscle involved in respiration, but we have the ability to take exaggerated breaths. And that ability to take exaggerated breaths is what our accessory respiratory muscles do. We have already talked about some. Our sternocleidomastoid and our scalenes help us with exaggerated inhalations. Our pectoralis minor, exaggerated inhalations. Our abdominals, which we'll talk about, play a role in exaggerated exhalation. Our serratus anterior compresses the chest for an exhalation. And the internal and external intercostals help in inhalation and exhalation. And it would be super convenient if our internal intercostal was involved in inhalation and our external intercostal was involved in exhalation. That would be incredibly, incredibly convenient, but it turns out to not be the case. Instead, it is the internal intercostal that helps us to exhale, and our external intercostal that helps us to inhale. So you just have to remember that they're the opposite. The E external, I inhales, The I internal, E exhales. So they do the opposite of what we would expect them to do based on their names. But basically, the internal intercostals bring the ribs down, and the external intercostals bring the ribs up to increase the amount of air we bring in or decrease the amount of air we bring in. All right? Questions on that? All right, I make a point of emphasizing this here. And again, I'll go ahead and save this pretty picture so I can clear it. Because the next muscle group we need to talk about are the abdominal muscles. Or more specifically, the muscles that form the abdominal girdle. A girdle is a support structure. And that's what your abdominal girdle is. It is muscles providing protection for the organs of your abdominal pelvic cavity. We talked about how your ribs protect your heart and your lungs. A little bit of your liver is tucked up there. A little bit of your kidneys are tucked up there. But the majority of the organs of your abdominal pelvic cavity are not protected by bone. Instead, they're protected by lots of different muscles at all sorts of different orientations. The best example of this is one of the most common abdominal muscles that we think of, and that is the rectus abdominis. The rectus abdominis, and I'll just draw 3 rows of fascicles here. All of the abdominal muscles are related to the orientation of their fascicles. And so here we have the rectus abdominis. With the rectus abdominis, rectus means straight. So this is the straight muscle in the abdominal region. And these fascicles basically run in a vertical orientation. One of the interesting things about this particular muscle is that while all the fascicles are parallel, there are actually tendinous insertions that actually divide the rectus muscles into 4 individual bellies. So we have 1, 2, 3, 4, 5, 6, 7, 8 individual abdominal bellies, and those are what we commonly think of as our 6-pack, or what is really in reality an 8-pack. One of the interesting things about these individual bellies is each one of these individual bellies have their own innervation. So it's actually possible to just contract one of these bellies. Now, just because technically it's possible to do it, doesn't necessarily mean that you can do it. The same way that not everybody's capable of wiggling your ears. You have muscles that are capable of elevating and depressing your ear, and some people can consciously control them and wiggle their ears, and some people cannot. And it's the same thing here, and probably one of the greatest examples is Terry Crews. For those of you who have seen Terry Crews, he's got some great YouTube videos where he actually has a voltmeter set up to him, where when he contracts the muscle, contracting that muscle generates an electrical stimulus, and it plays a musical instrument. And so he plays all these different musical instruments just by contracting the muscles of his body. So he has control of that. Other people can. It's possible too. You may. But we have that 8-pack that is formed by the rectus abdominis muscles. The next muscles, actually, before I talk about the next muscles, the other thing we need to talk about for our muscles are their actions. Remember, our muscles are always pulling across joints, or at least that's the goal, is to have them pull across the joint. In the case of the rectus abdominis, the joint that it's pulling on might not actually be obvious. However, what it's actually pulling on is it is actually pulling on the vertebrae. So what we have is the vertebrae of the back, the thoracic cavity and the lumbar cavity, that those are pulling on. So when these muscles contract, basically what they do is they flex the vertebral column. All right, so if you were doing a sit-up, for instance, that flexing of the vertebral column, bringing your head towards your knees, would be an example of one of the functions of the rectus abdominis. Now remember, the other function as the abdominal girdle is to provide protection. when we contract all of these muscles at the same time, when we contract these muscles, they tighten up the abdominal region to provide protection. Again, you would think of it if someone was getting ready to punch you in the stomach, you would contract that muscle, you'd compress your abdomen, and that would provide protection. And it's very, very strong. In fact, essentially, that's how Houdini died. Harry Houdini, as many of you know, was a very famous escape artist. And he would put on these huge, massive shows where they would have these elaborate contraptions that he would get put into, like, underground tanks or sand pits or all these types of things. And then he would, get out of the straitjacket, get out of the handcuffs, get out of these things and escape. And the setup of these things was incredibly elaborate and took a long period of time. So often when he would do these shows, he would do 2 or 3 of these escapes, and then they would need filler while they were resetting the stage for the next act. Some of the things he would do is he would have other minor acts come up and, like ventriloquists or dancers or things along those lines. But one of the things that Harry Houdini was known for was his constitution. And so what he would do is he would stand on stage and he would goad and he would chide and he would, he'd crack smart to a bunch of the men, and he would encourage anybody who wanted to come up on stage and punch him in the stomach. And they'd punch him in the stomach and he would stand it and he'd laugh at them and he'd call them girly men and all those kind of things like that. And he had this amazing constitution with his powerful core muscles, and he'd have lines of people coming up to punch him in the stomach. And that was part of his show. Of course, he was able to prepare for it because he would be able to contract his abdominal muscles and protect himself from that. So one day after performance, he was walking out of a bar. And as he was walking out of this bar, a couple of college students who had been at the show the night before happened to be there and see him coming out. And without any warning and without saying anything at all, they punched him 3 times in the stomach. And as they punched him in the stomach, they ended up rupturing his spleen, and he died a couple days later. So the famous escape artist Harry Houdini died from an unprotected, unexpected blow to the stomach. Because again, he wasn't able to contract those muscles because he didn't know it was coming and he didn't have that protection. So again, if you think of the relationship, our rectus abdominis run parallel to our vertebral column. So basically 2 actions. All of our abdominal muscles can compress the abdomen to provide that protection, but our rectus abdominis also can flex the vertebral column. Our other abdominal muscles are all lateral to the rectus abdominis. And again, they're going to be different in their orientations. The next one we'll talk about, and we'll go ahead and make this one red, is perpendicular to the rectus abdominis, or horizontal in its orientation. If you think about it, this horizontal orientation is similar to what we would think of as a transverse section of the body. So not surprisingly, it is called the transversus abdominis. These muscles are the deepest of those lateral muscles. And like I said, they are horizontal in their orientation. Notice, remember, our goal is for this muscle to affect a joint. However, if this is a horizontal muscle, it really doesn't cross one of the joints of the thoracic vertebrae or the lumbar vertebrae. However, if you think about it, this one basically comes around to the one on the other side. And in many ways, essentially what our transversus abdominis muscles are simply a circular muscle. The muscles from the 2 sides basically form a circular muscle. And as we know, when we contract a circular muscle, the lumen gets smaller. And so if you notice, our transversus abdominis really only has 1 action. It can't affect the vertebral column, but what it can do is compress the abdomen. So it has 1 action. Our rectus abdominis has 2 actions. And that leaves us with our last 2 muscles. The last 2 muscles are the internal and external oblique. All right. These muscles sound similar to the internal and external intercostal because they are going to be similar in their relationship. Our internal intercostal, internal oblique is obviously going to be deeper. It is also going to, like the internal intercostal, its fascicles are going to project down and out. But again, there's a third difference between the internal oblique and the external oblique. And here the internal oblique are more inferior. They actually stop at the lowest ribs. So if we were to draw these, again, their fascicles go down and out. And the same thing on this side. Whereas our external, and what color haven't I used? Let's use yellow. Oops, external oblique. Oops, no I. Because they're external or superficial. Their fascicles, like the external intercostals, project down and out, down and in. And these are more superior. They actually cover the lower ribs. And they actually interdigitate with the serratus anterior. So they basically come up to and basically intermingle with the serratus anterior. So those, and I'll cheat and put this down here for right now so that I can draw my lines. And again, these are going to come from more superior position, and they're going to come down and in. Sneak that up there. And the same thing on this side as well. Oops, there. So down and in. All right. Now, that is the orientation of this, and we'll look at the pretty picture that goes with this in a second. But there's one more thing that I want to point out to you, and I'll use an arrow to emphasize this, and I'll use black to show this. Notice when it comes to the functions, notice that because of the way these are oriented, the orientation of the external oblique on one side is identical to the orientation of the internal oblique on the other. So when you look at the, and I'll answer your question about the rectus sheath in just a minute. When you look at the actions of these two muscles, notice they have the ability to have actually both have 4 actions. And what do you notice about the 4 actions of the internal oblique and the external oblique? When you read them on the list, what do you notice about them? They're the same, exactly. They're identical. Because basically what one does on one side, what the external oblique does on one side, the internal oblique does on the other. So if you think of the 4 actions, and again, I think if I cheat here, need a little more light to make this work. If you think of terms of the actions of this, right, both of them compress the abdomen, both of them flex the vertebral column, right? So those are the same as the rectus abdominis. But because these are angled muscles, right, going at angles on one side, what they also have the ability to do is to rotate your vertebral column, and they have the ability to lateral flex your vertebral column as well. So these have 4 actions: laterally flex, regular flex, right, compress the abdomen, and rotate the vertebral column. So we have those 4 actions of these muscles, of these 2 muscles that do those things. All right, I, again, have done this picture of it here, but let's go ahead and look at the, come on. And then when I look at the picture, the pre-picture from your textbook, I'll be able to answer that question about the rectus sheath as well. So we did that. These are the internal and external intercostals. We did those. We talked about the levator ani. Here we see the abdominal muscles. Notice here are the 4 bellies. Let me get my annotation tool back and my spotlight. Here are the 4 bellies of the 2 rectus abdominis. Notice the fascicles are going in the vertical orientation with 3 tendinous insertions. So we have 4 individual bellies. Over here laterally, we can see how they've peeled away some of the muscle layers. Look over here. Notice, and again, the first thing we want to look at is the fascicles. These fascicles are coming down and in, and it's on top. This is the external oblique. Notice the external oblique comes up and interdigitates with the serratus anterior. So it comes up over the ribs to the serratus anterior. Notice here we've peeled away part of the external oblique, and we see that the internal oblique, whose fascicles are going down and out, only come up to the ribs and stop. And then deepest of all is the transverse abdominis, where they're horizontal, and they basically form a circular muscle all around. This is a fun way of seeing this, and we'll come back to this in a second. But here's what I also want to show you. I love these pictures from your textbook because they do this great job of isolating the individual muscles. So here we see the external oblique fascicles come down and in, goes up more superiorly on the ribs, over the top of the ribs, where it would meet the serratus anterior. Whereas here, we can see our internal oblique, the fascicles go down and out, and it barely comes up over the top of the lowest ribs. Here we see the rectus abdominis, which is vertical. Here we see the transversus abdominis, which is horizontal. So we can see these individual muscles. Now, we are not responsible for the origins and the insertions of these muscles. But the other thing that you can see here is this structure right here. This structure is an important ligament that goes from the superior anterior iliac spine to the pubic tubercle, right? Those are 2 of our bone features we had to learn. And this important ligament, because it goes bone to bone, is called the inguinal ligament. Notice if we go back to the previous picture, come on, go back to the previous picture. Here we see that inguinal ligament again, going from the superior anterior iliac spine to the pubic tubercle. As you can see, it's an attachment point for all of the abdominal muscles, but like I said, you don't have to know that. But the other thing that's on here we need to know is in that inguinal ligament is a space called the inguinal canal. In female type people, they have a potential space here for the inguinal canal, but it's not a true opening. Nothing passes through it. In boy type people, a structure called the spermatochord passes through it. Because in boy type people, our genitalia, our gonads, are located on the outside of the body for temperature regulation. where we produce our sperm. There are some biologists who think our sole function as organisms are to produce offspring. So if that's your sole primary goal as an organism, then putting your gonads where you produce the gametes to make those offspring outside of the body seems pretty crazy. Yeah, you have the potential to win $10,000 on America's Funniest Home Videos if you get hit in them. But again, that doesn't necessarily seem to make it worthwhile. But we stick it out here for temperature regulation. And so not only do the sperm have to get back into the body, but the blood vessels, the nerves, and the sperm have to pass through a tunnel. And that tunnel is surrounded by a structure called the spermatocord. And that spermatocord goes through that inguinal canal. This is significant, and I'm going to cheat and go back one more picture to this one here, where again we see that here's the opening to that inguinal canal, and here's the spermatocord coming through it. I make a point of emphasizing that females don't have a structure that passes through here and males do, because this is a weak point to the abdominal pelvic cavity. If the pressure increases too greatly in the abdominal pelvic cavity, these muscles are very powerful muscles, and as we're squeezing the organs on the inside, because you're afraid you're going to get punched in the stomach, or you're trying to lift a heavy box, or you have a defecation that is particularly difficult, we contract these muscles. And what can actually happen is if you put too much pressure on your abdominal pelvic cavity, the pressure will look for a weak spot to come out. And what can actually happen here for males is fluid, or even in some cases the omentum, the mesenteries, or even like a piece of the small intestine, can actually bulge out of that weak point from an increase in pressure. What do we call that? Anybody know what we call that when that occurs? Absolutely, that is a hernia. And in this case, for males, it would be an inguinal hernia. Because this has something that goes through it, this is much more of a weak spot for males. So inguinal hernias are much more likely in males than they are in females. It can happen in females, but very, very rarely. But there is another weak point where both males and females equally have the opportunity to have a hernia. What's that other weak point that both males and females have? I'll ask the question again. What is that structure that all males and females have that is a weak point in the abdominal pelvic cavity? The belly button, absolutely, the inguinal region and the umbilical region. And so it is possible to have an umbilical hernia, where you can get the bulging in the belly button region as a result of that. All right. Now, there's one more great picture that I want to show you. I love this picture because it shows them isolated, but I also love this picture, and this is where we'll finally get back to Ashley's question, of the relationship in a cross-section. Notice again, here we have the rectus abdominis muscles, and here we have the three lateral muscles. Deepest is the transversus abdominis, then the internal oblique, and then superficial is the external oblique. But notice, like all muscles, they have tendons that come off of them. Notice all of them have tendons, but it is the external oblique tendons that's on top. So when we go back to this picture, this big, broad, flat tendon we see on top is the aponeurosis. Remember, aponeurosis is the fancy word we have for a broad, flat tendon. This is the aponeurosis of the external oblique. But if we look closely at the picture, we can see that the tendon from the external oblique, the aponeurosis of the external oblique, and the aponeurosis of the internal, and the aponeurosis of the transversus all wrap around and stabilize the rectus muscle, forming a sheath around it. So if you notice, that rectus sheath is basically formed by the aponeuroses of the three lateral muscles. And what's cool is these collagen fibers come off of the muscle, wrap around the rectus abdominis, and actually come together with the collagen fibers on the other side. Notice right on the midline between the two bellies, and here we see it, but we also see it nicely on this picture. Right in between the two bellies, the collagen fibers fuse together to form a white line down the middle. And that white line down the middle is Jessica's younger sister. It is the linea alba. So linea alba literally means white line, like albino, linea, line. It's the white line down the midline of the body. So down the midline of the body, where those collagen fibers, those aponeuroses all come together, we have that linea alba. And so here we see it here as well. All right. Oops, too far. Questions on that? Linea alba gets excellent. Absolutely, absolutely. So that, let's go back to this pretty picture. I wasn't going to mention that, but you're absolutely correct. One of the things that happens during pregnancy, when that basketball is growing inside of the female, as the muscles are pulled further apart, this especially occurs inferior to the belly button, but can occur above it. That collagen fiber gets stretched, and as it gets stretched out, it thins. As it thins, it becomes more transparent, and you can start to see some of the darkness of the inside. It's not like you can see the baby kicking around or anything, but the whiteness of that line goes away and it becomes darker, at which point it's called the linea nigra, right? The black line. And so it is a common feature that can occur during pregnancy. And then typically once the basketball is expulsed, often, again, because the decrease in pressure, the decrease in size, the linea nigra goes away as a result of that. And so our linea alba comes back. So absolutely. So that occurs because of the stretching. All right. Excellent. Speaking of which, there's one last structure. So linea alba is a structure, aponeurosis of the external obliques is a structure, inguinal ligament is a structure. The last structure we need to identify, oops, I guess it was the wrong direction, is this one here. Notice on the front, those are aponeuroses because they are specific tendons of the muscle. Here, what we have, this triangular region, and I know it looks like a broad tendon because of the way they've drawn it, but you have to just trust me, this is artistic license again. What we have here is a connective tissue that is stabilizing numerous muscles here in the back. Because it's stabilizing numerous muscles in the back, it isn't a tendon, it isn't an aponeurosis, it is a fascia that is stabilizing many muscles, especially the muscles of the spine, which we're not seeing here, and more importantly, you're not responsible for on the exam. But it is a fascia in the lumbar and thoracic region of the body, which is why it is called the thoracolumbar fascia. So this white, a diamond-shaped region on the posterior part of the body is the thoracolumbar fascia, and that is the fourth structure you are responsible for in the trunk region. All right. Questions on any of that? All right, I think in that case, we've actually pretty much hit all of the trunk muscles. And so again, if I've forgotten any, again, you're still responsible for it, but I think we've pretty much hit all of them. The last thing that I want to do, how are we on time? We're doing good, is that I want to talk a little bit about the arm muscles and their relationship. And we'll talk more about this on Thursday. We'll get more of it on Tuesday next week, or not next week, but in 2 weeks as well. But I want to make sure, because there's a couple things here I really want to emphasize, to make sure we understand the relationships of these multi-headed muscles. The first example of a multi-headed muscle is the biceps brachia. As we talk about, there's often a lot in a name. Brachia obviously means the arm region, and biceps literally means two heads. And one of the important things to remember is if we have multiple heads, we have multiple origins. All right? So if we have multiple heads, we have multiple origins, and you are responsible for both individually. I will remind you of that by using the key phrase on the exam, be specific. If you see that term, be specific, that doesn't mean tell me everything you know. It means tell me precisely the piece, the singular piece of information that I want to know. Let's cheat and find, start with black. What we have here is our bicep brachia. It is a two-headed muscle. I know our illustration doesn't do the best job of showing this, but the models do a nice job. Other things do a nice job as well. And what we have here are the two heads, two bellies, and they are side by side. So the first belly is basically this portion right here. That is the first belly. And the second belly, let me change the colors. Now what else will stand out well? I think the green will stand out well. This here is the second belly. So we have two bellies side by side. And on the exam, I may have a tag on one. Or I may have a tag on another. Oops. And you are going to be responsible for distinguishing them. So on the exam, if I ask you to identify the muscle indicated by tag 1 and identify the muscle indicated by tag 2, and again, on both of those questions, I will say the key word, be specific, and I will tell you right now that the answer to those two questions are different. All right, let's give myself a little bit more playing room. So let's cheat. I'm going to make a big white box, and with that big white box, I'm just going to cover that all up. Excellent. Yeah, let's cover all of it up. That'll give me some more playing space. So in this case, again, identify the muscle, and I will say that keyword, two words, be specific. Oh, I have my cap locks on. Identify the muscle indicated by tag number 1. Be specific. That is exactly how I would ask the question on the exam, or I could ask the exact same question again and say with 2. So let's ask that question again. Identify the muscle indicated by tag number 2. Be specific. When you see that phrase, be specific, that is when your warning bells should go off that something special is going on with this question. Typically, what it means when I say be specific is that it is a multi-headed muscle, and you need to be able to distinguish these. The bicep brachia has two heads. One is the long head, and one is the short head. So if I ask you to identify the muscle indicated by, that should say tag and not tag B or whatever that is, be specific, and I ask you 1, you do not want to write that the correct answer to that is the short and long head of the bicep brachia. If you write that, you will get it wrong because you were not specific. The specific one that is tagged by number 1 is the long head of the bicep brachia. The precise head that is indicated by 2 is the short head of the bicep brachia. And again, you don't have to put of the, just put short head, biceps brachia, and that would be fine. Doesn't have to be a complete sentence. But again, I'm not asking for both. Can you tell me 2 seems longer? Yes, I will exactly explain to you why that is the case. And I know it is a little bit confusing as to why they are. The key with these and the way you distinguish the short and the long, and let's do this. What really determines the short and the long is the length of the tendon. It's not actually the muscle itself that determines whether it's long or short. It's actually the length of the tendon. notice the tendon for the short head basically comes right here from the from off of the muscle, stays anterior, and comes straight up to the coracoid process of the scapula. Whereas the long head, right, the belly looks a little shorter, but its tendon actually comes up through the intertubercular sulcus between the greater and lesser tubercles, up the intertubercular sulcus, over the head of the humerus into the joint cavity, and goes all the way to the supraglenoid tubercle. So it's a deep tendon. We don't see it all, and it actually goes into the joint cavity, but it is a much longer tendon, and it's that longer tendon that makes this the long head. The other thing that helps me to remember this is that the long head is lateral, and the short head is medial. So again, medial short doesn't help you, but lateral long, L for long, L for lateral. It is the lateral head that is long. So when you see that magical term, be specific, that tells you I want a specific piece of information. In this case, you need to be able to specifically distinguish the two heads. However, there is a second thing you need to be able to specifically distinguish. Not only do we have to tell the two heads, but we also have to tell the two origins. So again, in this case, you could also see the potential question, identify the origin of the muscle. I didn't really want to use green, but that's all right. Indicated by tag number 1, be specific. Or I could ask the question, identify the origin of the muscle indicated by tag number 2, be specific. And again, when you see those two questions and that magical phrase, be specific, you know again that the answer between these two questions would be different. If we have a different heads, we have different origins. And so, of course, the answer, as we already indicated for 1, the long head, as I mentioned, is the supraglenoid tubercle of the scapula. And again, I want bone and bone feature. A lot of you lost partial credit on the lab exam because when I asked for bone and bone feature, you did a great job of answering the bone feature part, but you forgot to say the bone. Many of you gave me the harder of the two answers, but if you don't give me both, you don't get full credit. So the origin of the long head specifically is the supraglenoid tubercle. The origin of the short head specifically is the coracoid process of the scapula. Again, if I gave you this question right here on the exam, and on the exam in the space that was there, you wrote there in a full paragraph worth of information, the origin of the long head is the supraglenoid tubercle of the scapula, and the origin of the short head is the coracoid process of the scapula. All of that information is 100% correct, but you are not specific to the tag. And if you're not specific to the tag, you will not get it right. Even though what you said was 100% correct, you do not answer the question, you cannot get full credit. So it is very, very important too. And again, I will give you that magical, mystical phrase. Oh, that didn't work. That magical, mystical phrase. If you see the magical, mystical two words, be specific, that is a dead giveaway that I want precise piece of information. And so make sure you provide it for me. Now, the good news for the bicep brachia is that while it has two heads and it has two origins, it just has one insertion. So if I ask for the insertion for A and the insertion for 1 and the insertion for 2, the insertion is going to be the same. And also the actions are the same. The action of 1 and the action of 2 is the same. So if I have either of those two tags, and I asked you to identify the insertion, or I asked you to identify the action, for those, they would be the same. So I wouldn't say be specific for those. So notice I'm only going to use be specific for origins and for names, because that's where they're different. The names are different, the origins are different. The insertions and the actions are the same. It's an important thing to remember when you're studying this. So notice the bicep brachia has two heads that need to be identified and two specific origins. Guess how many heads the tricep brachia has? 3. And so guess how many origins you're going to be responsible for? 3, absolutely. So again, you have those lists there. So again, I want you to work on these things. I want you to think about these things. I want you to stand up and I want you to do the actions, right? See what it is to use the deltoid, to abduct the arm, to medial and laterally rotate it, right? Try to figure out, like I said, what these things are used for. And again, standing up and using these muscles and seeing how they move your body is a great way to remember how they function, right? Let's look at the bicep brachia. That's what I want. When we look at the bicep brachia, as we've talked about, we need to be able to identify the origin and the insertion. So let me get my line. Let me get my red, because that works. We know the origin for the short head is over here. So again, I can draw my line up here. This is the short head on the coracoid process. And the cool thing about my drawing thing is I can use pink, which is almost red, to emphasize that up here on the top of the glenoid cavity, that bump on the top of the glenoid cavity, is my origin for my long head. And I know that both of them insert into the radial bone, the radial tuberosity. So that is the insertion for both of them. So notice, like we've talked about, we're able to draw the origin and the insertion. And the cool thing about this, as I've talked about, is it helps us to understand its function. Notice muscles move across the joints that they cross. Notice in this case, we cross the elbow and we cross the shoulder. Our bicep brachia is an anterior muscle. One of the important things is, and again, we always have to throw that word most, because there is one exception. The one exception is the knee. But if you think about it, remember, all of our flexes are anterior movements, with the exception of the knee. The knee is the only thing that when it flexes, it goes back. So what that means is most anterior muscles are going to flex. So our bicep brachia is an anterior muscle that crosses the shoulder and the elbow. So when we think of functions, it is going to flex the shoulder and it is going to flex the elbow. And again, remember, instead of shoulder, I could say flex the arm or flex the forearm. Either of those are acceptable. But here's the other key, and let's switch colors again. I don't want that one. Let's go with green. When we're on the forearm, remember our forearm has that ability to do that funny thing where it can pronate and supinate. So hopefully you guys remember pronation and supination. If you remember, when we did that pronation and supination, both forearm bones didn't move. If you remember, the ulna was stationary. and it is the radius that rotates around the ulna. This is significant because when we come over here and look at our insertion, we insert it onto radius. So that's the movable bone, not the immovable bone. So if a muscle attaches to the radius, then it's able to either supinate or pronate. Right? It's going to play some role. If it's on the movable bone, then it can supinate or pronate. If, however, a muscle attaches to the ulna, since the ulna is stationary, bone muscles that attach to the ulna cannot pronate or supinate. Our bicep brachia attaches to the radius, the movable bone, and its function is to supinate. Let's actually do that so it's more out of my way. Excellent. And there, just by looking at the bones, seeing where the attachments are, we can think of the activities that our bicep brachia does for us. Now, you may not have thought of it in these terms, but hopefully looking at this helps. But the other thing to think about, too, is to think of the action of it. I keep making the joke about doing the curls for the girls, right? Because you do it to get that nice, big, superficial bicep brachia muscle that is a superficial muscle that sits on top of the arm. All right, when we're doing those curls for the girls. Now, when you do those curls, typically you are doing it with your hand in the supinated position, right? You're doing that with your hand, and again, I'll cheat and turn the video back on, right? You're doing this with your hand supinated. So as you're lifting weight, you're lifting that weight, doing those curls for the girls. Is it possible to pronate and still flex the elbow? Do this type of exercise? Absolutely. Can you lift more weight pronated, or can you lift more weight supinated? Yeah, more weight supinated. Because when you pronate, what you're actually doing is twisting up that tendon of the bicep brachia. So when you're pronated, you can't actually use your bicep brachia as efficiently, and you can't lift as much weight. But when you're supinated, the way it moves the hand, then you're actually able to lift more. So we can actually see that in its action. Now notice, as we talked about, it isn't the only muscle that is responsible for flexing the arm. Deep to your biceps brachia is a brachialis muscle. I'll show it to you in a minute, but I want to draw the origin and insertion first since we're here. If we think of the origin and insertion of the brachialis is right underneath the bicep. I'm going to cheat and draw them over here. So here's the bicep brachia with its two bellies side by side, right next to each other, superficial. And deep beneath them, again, obviously this is exaggerated by size, but deep underneath them, directly underneath them, is the brachialis muscle. So they're sitting right on top of each other. They look right next to each other. However, when we look at the origin and the insertion of the brachialis, we can see how they're similar but different in their functions. The origin of the brachialis, as you can see, is basically the diaphysis of the humerus. So basically it's over here on the origin of the brachialis is the distal half of the humerus over here. It actually, let me cheat and change colors here. Our deltoid, that lateral muscle, comes here on the side and points right at it. So it's here. And then it inserts into the coracoid process of our ulna. Notice the brachialis and the biceps brachia are right next to each other. But notice the biceps brachia starts up here on the scapula. So it can affect the shoulder where the brachialis cannot. The bicep brachia attaches to the radius, the movable bone, so it can supinate, whereas our brachialis attaches to the ulna, the stationary bone. So it is not able to supinate or pronate. And so if you notice, it's an anterior muscle. It just crosses one joint, and it only has one function. Its one function is to flex the elbow. All right? Now that we see where it attaches, when we go back to the pretty picture, here we see the relationship. Again, here is the bicep brachia with its two bellies. And right underneath it, deep behind it, right here, is the brachialis. Although notice the bicep brachia goes to the radius, brachialis is going to the ulna. So again, notice taking the time to draw the horizontal insertions can show us how two muscles that are very similar in their location can be very different in their functions. Both flex the elbow because they both cross the elbow, but the bicep brachia also crosses the shoulder. It can affect the shoulder. It also attaches to the movable bone, so it can supinate the arm, whereas the brachialis is on the stable bone and cannot pronate or supinate. Now, one last thing. There was another term, an important term on that previous page, and that is, or on the instruction, embracing. Basically, what embracing means is that when one muscle begins, another one ends. The deltoid's a great example. The trapezius, if you remember, comes into the lateral part of the clavicle, the scapula, and the spine of the scapula and the acromioscapula. And that is actually the origin of the deltoid. So those two muscles embrace. Another two muscles that embrace are the brachialis and the deltoid. The reason this is important takes us back to our bodybuilder pictures. Notice again, we see the massive exaggeration of these muscles. But here we can see this relationship. Notice here is the trapezius, and where it inserts is where the deltoid begins. So here we see the deltoid muscle coming down. But look at this one in particular. Our trapezius comes here to our deltoid where our deltoid starts. And our deltoid inserts right here. And notice where it inserts is right next to the brachialis. It points. No matter how big these muscles get, the deltoid is always going to point at the brachialis. Now, normally this does this on the side. Notice he has his arms turned inward. But with his arm turned inward, we can actually clearly see this deeper muscle, the brachialis. Again, remember I told you are going to be responsible for recognizing muscles on bodybuilders, but it's just going to be superficial muscles, except for things like this. The brachialis technically is a deep muscle, but because the deltoid embraces it, it points right at it. So here we actually see the long head of the bicep brachia. Here we actually see the lateral head of the tricep brachia. We haven't identified that, but it's on the lateral side, so that's not surprising. And then right in between them is the bicep brachia. So one of the nice things about these bodybuilders and their definition is we can clearly see the individual muscles. And so this is indeed a deep muscle, but because the deltoid points at it and embraces it, oops, go back. Whoa, too far. It is a deep muscle, but because the deltoid embraces it, it is one you'll be responsible for on the exam. All right. All right. So that hopefully is a good starting point for this. Deltoid points to the brachialis. Yes, that is correct. And here we see it on the non-human example. Here we see the very cut tip of the deltoid, and notice it points. The point of it points right at the bicep brachia. Here's a great place where you see this. Let's go. No, hold on. What I want to do is here. When we go to the limbs, perfect. Notice we are looking at the posterior part of the arm here. This here, again, when we're talking about the tricep brachia, we didn't talk about it, but I want you to pay attention to it. It's easy to find because it's the only really posterior muscle on the upper part of the arm. And here, it has one big tendon that all three bellies connect to. This is one belly, the lateral belly that connects to it. This is the long head or belly. And then this tiny thing down here that you can't really see, but we'll see in other views, is the medial head. So medial, lateral, long, those are the three heads of the bicep brachia. But what I want you to pay attention to is this right here. This triangular muscle is the deltoid, and that arrow points, boom, right at this muscle. This is the brachialis. So that is the bicep brachia. In fact, that's the long head of the bicep brachia because the long head is lateral. There's the lateral head of the tricep brachia. The lateral head is lateral. And that deltoid is a big arrow that points right at that deep muscle that is the brachialis. So notice there is our tags from that study guide on the visual anatomy thing from CRC. So we can see that really, really nicely. Our models show that excellently. All righty. So again, lots of ways to study this material. I want you to look at these pictures of the models. I want you looking at the pictures from your textbook. I want you looking at all of these resources and these things. But I also want you to take the time to draw the origins and insertion. And I want you to do that for two reasons. The first reason I want you to do it is because seeing how these muscles attach across the joints can really help you to understand how the muscle functions. But the other reason I want you to take the time to draw the origins and the insertions is because on this exam, again, usually when it's a 30-station exam, anywhere between 4 and 8 of those stations. So if you think about it, 25% of the stations could be bones. So I could have a bone like this one here, and I could have a tag on it. And again, let's go ahead and use something like purple. I could put a tag right there on the zygomaticus bone. And again, it would be a real bone picture, not a drawing like this, but I could have a skull. I could have a bone feature of that skull pointed out. I can say identify the muscle that originates from that bone feature. And you need to remember that it's the zygomaticus that makes you smile that originates from there. Or I could take that same tag and I could put it on the mandibular angle and ask you to identify the muscle that inserts into that bone feature. And you would, of course, know that that's the masseter that inserts there, and so on and so forth. So I will show you pictures of bones, I will tag bone features, and I will ask you to identify the muscle that either originates from or inserts into that bone feature. And one of the fun things about that is, like, for instance, this coracoid process of the scapula. As we just learned right now, I could ask the question, identify the muscle that originates from this bone feature, be specific. And you would, of course, answer that is the short head of the bicep brachia. But if you also remember, I could have that exact same picture with that exact same coracoid process labeled, and I can ask you to identify the muscle that inserts into that bone feature. And the muscle that inserts into that bone feature is the pectoralis minor. So some of these bone features are origins to some muscles and insertions to others. So spending the time with the bones will help you to remember that, because somewhere around, my guess for this test, probably somewhere between 10 and 20% of the questions will be bones, where you need to be able to identify what muscle would insert or originate from a particular bone feature just by looking at the bones. Now, we talked about this when we were doing the bones. We're going to learn them once and use them twice. Well, here we're going to be using them again. So my guess is somewhere between 10 and 20% will be that. All right, so that's your game plan. Remember, for Thursday's class, your unit review is due. For Friday, make sure you do the practice exam. Make sure you do the muscle activities. And again, even though you're not turning in these bones and bone features, I strongly encourage you to learn them. They will help you tremendously. Make sure you're taking advantage of the resources. Again, that personal anatomy lab where you can look at the animations and see the individual muscles and what they do is a great, great tool. So make sure you're spending the time with all of these things and learning this material. All righty. Any questions on any of that? All right, excellent. I will leave you guys to it. I encourage you to spend a lot of time between now and Thursday so that you can ask questions on Thursday. You have a good day. As always, stay safe, stay healthy, take care of yourselves, and I will see you all soon. I'll stick around for another couple minutes or two just to make sure that you guys don't have any other questions that I can answer, but I am going to stop the recording, and then in a couple minutes I will shut down the class because, again, I remember I have to close the class so that the video will can be converted and I can post it on YouTube, and it'll be posted this evening. All right, guys, last chance. Going once, going twice. All righty. Have a good day.